Wonderful good morning to everyone, wherever you may be, online or in the room. I'm very happy and honored to open this meeting. Let me shortly and immediately broaden the topic. The topic is sarcomas, the topic is rare sarcomas, but what we have here, what we intend to do today, goes beyond the specific issues of very rare sarcomas. Where we start from, or let me make that a more personal view, why I think that is very important what we do here is that because we, some colleagues and I, perceive a gap between the clinical practice on one hand, what you, dear colleagues, do outside there with the patients, and the regulatory decisions we have to do on the very medicines you use. And this gap is not only in the practice, there is also some unfulfilled expectation each side has on the other one. We do have on the clinician practical side the strong dominance, the very great awareness of the unmet medical need, of the need to treat, if nothing else is there. Very patient-centric. And we have to admit, on the regulatory side, we do have the legal framework, the regulatory framework we have to stick to, that is written down, that has been written down sometimes many, many decades ago, saying, oh, you have to show this and that, and with this and that stringency. Mostly those rules, when they were made long time ago, were made with standard products, standard procedures, standard situations, standard diseases, if you like, in mind, and might not be very suitable, very helpful for more complicated situations. There are vectors, there is the exceptional circumstances regulation, but still there is obviously this gap between what we want and what we can do at the moment from both sides. And therefore, what we want to do here and today is to start a longer process. Yes, we will exchange some knowledge about things. That is good, but the more important thing is that we start here a process where we try first to exchange what we do, why we do it, and what we might do differently, and then start moving. Start decreasing this gap we have, getting closer together, and not only in the way we want things to be, not only in the way we think, but indeed what we do, how we decide, and how we act. That means for the regulators, what we want to start is to find better, newer, innovative ways to use the data that can be made. Not insist for any future on, oh, that is a strict rule, but to realize what can be done and see what are the best possible ways to work with what is feasible, what can be done. And from the side of the clinical experts, here it would start with knowing better what data, what information is useful for the regulators, what can be used, and then provide those as far as possible. So, bottom line, what I do hope for and what I wish for everybody, let's start something by exchanging news and information, and then end this day, end this meeting with clearly action points, with actionable points where we say that are the next step, and we are starting on a long way. Let's go clearly beyond, oh, it was very nice and interesting to have talked about it, to learn all that. Let's start a real process. And to allow that, I will stop talking now and leave the floor to those who are really in there. Caroline, back to you. Yeah.
Thanks a lot, Harald. Complete agreement. This is going beyond the topic for today, which is sarcoma. But sarcoma is the right model to discuss what we could do when we are facing orphan conditions, especially orphan conditions with severe prognosis and where patients are in very high need of an efficient treatment. And these are situations where we must be flexible, adaptive, I would say. And there must be some changes in the processes we operate normally for usual drugs where we have plenty of time before coming to a decision. The model must be adapted. So this is what we will discuss today, probably. I wanted to specify when we organized this workshop that this is not a collection of scientific advices. This is not something that will find solutions, not at that step, but we will try to build on ideas to find solutions. So my name is Pierre Demolis. I come from the French agency, as you could guess from the accent. So, I'm the system for, I've been working in the system for 20 years now, from the French agency with many European tasks. I was in the CHMP. I'm a member of the Scientific Advisory Working Party. I'm the chair of the Oncology Working Party. I'm a physician by training. That's it. But, okay, we will have to discuss what is exactly the European landscape, because I could notice that the academic world is a bit far from the regulatory world, or the regulatory world is a bit far from the other one. There is no responsibility at any side, but I think that some explanations to describe the landscape are necessary. So the EMA is a network, mainly. A network of national agencies of several countries in Europe. Some countries have two agencies, like Germany. Some have only one, like France. But we are all a network collaborating here in Amsterdam to come to proposals, not to decisions, because the decisions are normally taken at the level of the European Commission in Brussels. But we prepare these decisions, marketing authorizations, and so on. For that, we have an organization based on management board, a collection of people coming from the national agencies, an executive director, and because the EMA has to deal with human and veterinary medicines, here we are restricted to human medicines. We have committees group people coming from all the national agencies, group to discuss together, normally once monthly, and the CHMP is on the top of that organization. It is the first committee created at the European agency responsible for marketing authorizations, giving and removing and modifying the marketing authorizations. And other committees came a bit later. The PRAC is responsible for the pharmacovigilance. And for you, very important are the COMP, which is responsible for the orphan legislation here and its application at the European level. The PDCO for the pediatric legislation, most of you know already. And the CAT, created recently, who is responsible for the advanced therapies, genic therapies, cellular therapies, and so on. So this is a network, indeed, and the network has to discuss with external parties. So next slide, please. And because we do not, the next slide, yes, because we are not seen as an isolated structure, we must have discussions with our partners doing the same job, more or less, other regulators, the FDA. Hello to the colleagues from the FDA who were kind enough to join this meeting today. But other agencies, Swissmedic, the Australian system, the Japanese system, and so on. But we have discussions with other partners, and one of these partners, as an example, a very good example, is EORTC. We developed projects, discussions in common, and this is the natural way we should have the topics related to oncology dealt with at the European level, not only by the regulators and the external experts working for the European system. Within that European system, close to my heart, is the Oncology Working Party, The Oncology Working Party is a group of regulators who are interested in oncology and responsible for drafting the guidelines, giving advices. And the Oncology Working Party was very active in organizing this meeting. But thanks to the colleagues of the EMA, we were not alone to do the job. And a great thank to Caroline and Francesco here. So I have the honor of being the chair of this Oncology Working Party. The next slide, please, is a preparatory slide for the presentation Ralph held will do very soon. We have plenty of systems that could answer questions related to orphan severe conditions such as sarcomas. And this is throughout the whole life of a license, even before, but even after. And from these tools, we have, as an example, the scientific advices. But maybe the scientific advices as organized today are too a rigid procedure to answer the problems posed by orphan conditions. And just to let you know, we are reflecting at the Scientific Advice Working Party to a more flexible approach of the problems. We discussed that not later than yesterday. We have the guidelines. We have the Innovation Task Force in Prime, which are here to help when innovative projects are on the table. And we have some models of marketing authorization, conditional, under exceptional circumstances. But Ralph will explain all of that. So we have tools. The problem is how we can adapt the tools to the situation. And the other problem is how to use these tools, because the tools do not create the work. Somebody must operate the tools properly. The method is very important. And the method, which is applied, as explained Harald, for usual products, doesn't work for rare conditions or doesn't work satisfactorily. This being said, I believe it was my last slide. It was. So I'm giving the floor to Silvia Stacchiotti and the other chair of this meeting. Thanks for your attention. Thank you.
So good morning, everyone. I am Silvia Stacchiotti. I am a medical oncologist. I work at the National Cancer Institute of Milan in Italy. And I really would like to start by thanking EMA and in particular Dr. Pignatti, Pierre Demolis and Dr. Hensman and Caroline for helping organizing this meeting on behalf of all the colleagues from the EORTC and in particular the EORTC Soft Tissue and Bone Sarcoma Group, and on behalf of the colleagues who joined from the US, patient advocates, and also Anti-Cancer Fund and other non-for-profit organizations who are working with us. And on the other side, I would really want to thank all the colleagues who joined in person today and all those who are connected virtually and will take active part to the meeting. And I want in advance to thank all those who will try to make the best use of the material that is developed today to support in the near future the development of new treatment for patients affected by rare cancers and, of course, ultra-rare sarcomas in particular. Can I have the next slide? Okay, these are my disclosure. Next. Okay, I would like to start providing some background for the meeting that we have organized today. As of 2020, we have started working with EME as EORTC Soft Tissue and Bone Sarcoma Group on two different projects regarding ultra-rare sarcoma. Next, one project was about how to design prospective studies to develop immunotherapy in alveolar subpar sarcoma, and this project next was also joined by Anti-Cancer Fund. And next, the other project was on the repurposing of old drug in new indication. As EORTC Soft Tissue and Bone Sarcoma Group, we applied together with the patient advocates from the EHE Foundation, who are also here today, and Anti-Cancer Fund to discuss the repurposing of sirolimus in epithelioid hemangioendothelioma. These two projects help us make us very clear that how much important is for us working together if we want to fight discrimination against rare tumors, rare cancer, and in particular, of course, sarcoma. Next, sorry. And indeed, sarcoma is a family of more than 90 different malignant tumors that all of them represent a distinct entity. Next, and they are all rare, and some of them are extremely rare. Next. So rarity and heterogeneity mean paucity of data, and then of course major challenges when it comes to developing prospective randomized comparative clinical studies, and a lower level of evidence available compared to what we have for more common cancers. This translates into regulatory issue, and both the rarity of the diseases and the uncertainty in the outcome of the process that should bring to the development and approval of a new drug make rare tumors really less attractive for industry, especially when an agent is already approved in another indication. This translates in a few drugs that are under development, few prospective clinical studies, and eventually in a few drugs that are approved specifically for this indication. Then when a patient needs a treatment, often there are limited options, if any, and very often, unfortunately, the only way to access potentially active drug is represented by off-label agents. Off-label agents are not available at the same level everywhere, and this again translates into a huge disparities on how patients are treated across different regions and areas. So going to the going to the contents and the goal of the meeting today. Here today we want really to understand, to start working on how together we can create a novel methodology and new pathways tailored on the specific need of patients affected by rare cancers, among which, of course, ultra-rare sarcomas, and at the same time meet the regulatory requirements so that at the end we can really together be able to develop a new treatment option for our patient affected by these tumor types. So next, please. Okay. As a sarcoma community in 2019, we thought that an important point was to try to identify among all sarcoma which are those that are particularly suffering of this context, and in particular those in which rarity prevents the conduction of the classical prospective comparative randomized clinical studies. The group looking at the epidemiological data that are available and WHO classification identify in an incidence of 1 or less than 1 case per 1 million inhabitants per year the threshold below which all these problems are particularly relevant, and started calling these sarcoma types ultra-rare sarcoma. Next. We then made a list of all sarcoma who are ultra-rare, and we found that more than 75 different entities belong to this list, accounting for approximately 20% of all patients affected by bone and soft tissue sarcoma. And indeed, if we look at the prospective study that were ever conducted across all these entities, we can see that randomized studies were never performed, with the only exception being alveolar of sarcoma, in which we had 2 small non-comparative phase 2 study done in the past. On the other side, next, if we look at the list of the drugs that are specifically approved for this indication, in spite of the big number of different entities, we have only 4 drugs available for 3 different 3 of these tumors. Slightly more are available in the US next. And it's interesting to see that next 2 of these compounds were approved by an agnostic approach. So, of course, this drug adds to what is considered the standard of care for treatment of all soft tissue and bone sarcoma, represented by surgery, radiation therapy, and a few next systemic drugs that can be used for all sarcomas. Most of them are pretty old, and we have only one target agent, pazopanib, that can be used almost in all these indications. So next, so this is the framework, and we absolutely feel that we need to work alongside with the regulatory agency if we want to improve, if we want to make a better world than the one we could do in the last years. And for that, we conceived together with our EMA colleague the meeting today. The agenda, as you can see, foresees some initial talks to set the scene, not only for sarcoma, but for all rare cancers. Then after lunch, we will have a second session in which we will go a bit more into detail in ultra-rare sarcomas. This is not to talk specifically of these diseases, but to to present an example, a model that can be followed also for other rare tumors. And we will try to present what we have done, which kind of data are we currently collecting, to ask how can we improve in data collection and in designing clinical study. How can we create, as I said, a new methodology for this? Then after, we will be joined by our colleagues from FDA and NCI. And eventually we will have 1 hour for panel discussion in which we will try, of course, to answer at least some of the very many questions that we have. Today, we will also present a new global project from the sarcoma community that is called PUSH. PUSH was launched at the end of 2023 together with many other stakeholders, not only scientists, but also patient advocates, non-for-profit organization, and is meant as a project that wants to create a global consortium to maximize, on one side, the knowledge that we can gain from each single patient affected by ultra-rare sarcomas, and to make the best use of the data that we can collect to try to support development of new treatment to improve the outcome and quality of life of our patient. And we are here to say that we really would like to develop this consortium, this platform together. So we know that today this is only a starting point. This is the initial discussion, but we all know how much important it is to start. All our lives started from very important decision. There was a first day and then everything had to follow, but to start is important. And for that reason, as was said also before, we wish that this can be the first of several meetings in which we will be able to address all the single aspects that are inside these tasks. So thank you very much, and to EMA and all of you for being here. And let's start with the first presentation.
Thanks a lot, Silvia. So I believe we are in very good agreement on what we could expect from this meeting and from the following ones. Let's start the job. And the first presenter is Ralph Herold, who will explain how EMA deals with these rare diseases and what are the tools and organizations we have in hand to answer the questions. Thanks, Ralph.
Thank you very much, Pierre, and also Silvia. I'm both humbled and excited to be talking and interacting with you here. This is about practices for interactions. I'm trying to make a couple of links, not going into details of procedures or legislation, but trying to say where we are now and what are opportunities for engagement. And it will include an element of what we can do and an element of also what we find very difficult or cannot do. But I think the links are important for you to be aware of. Next slide, please. I would like to start with both kudos to the sarcoma community and a quotation that is more a problem statement. On the left-hand side, the kudos to the sarcoma community are in having managed an osteosarcoma EURAMOS trial that ran from 2005 to 2012 that had several thousand participants as an interregional trial, as a first such big effort that was made to bring together this rare, but among the bone sarcoma, still the most frequent bone sarcoma. So not ultra rare, not in this category, but still rare. I think we had only seen a few patients a year in my clinic. And this is an effort that has been made, bringing together 4 study groups on important questions and advancing the treatment with 2 randomizations in 1 clinical trial and producing results, allowing biological sample collection and so on. So I want to mention that this has taken huge effort. I believe this was well appreciated at the time and also is still a model. On the other hand, on the right-hand side, you see an analysis of the Accelerate multi-stakeholder platform that some of us founded together in 2017 and that you will hear about later today. But they have analyzed how clinical trials with children with the cancer developed over the past years into international and interregional trials, just as an update, so to say, to what I proposed to you with the EURAMOS, that was a trial of all patients of less than 40 years of age. And the Accelerate analysis says that only a fraction, maybe 9% and 5% are international and are interregional. And they are also saying that there wasn't much progress over the past years in having more international clinical trials with children with the cancer, whatever the phase and whatever the sponsor type is. So I think I take this as a motivation to look at this and hopefully contribute to improve it. Next slide, please. So what now we need to discuss if we think of getting new medicines to patients who obviously need it, because this is still our starting point, the patient needs and the willingness to work together for better outcomes and for improving patient lives and experiences. Next, please. The fundamental elements are on the left-hand side with the PRIME scheme, orphan designation and conditional marketing authorization. We have at the last occasion, and that was at the Beating Cancer Special Committee, set out these serve best for achieving to provide new treatments to patients with the cancer. To go briefly into this, PRIME scheme is a scheme where we are looking at early data from medicines and recognize there can be priorities to address unmet medical needs. And then there is a deep scientific advice, frequent scientific advice provided, and a continuity of interactions planned so that there is coaching of such priority medicines towards authorization, towards access for patients. The second fundamental tool for supporting new treatments is the orphan designation. Since we're talking here about rare diseases, it is the legal provision of the orphan designation that provides us with the ability to finance and to undertake additional targeted support to medicine developers. This starts from the orphan designation, which already can be seen as a scientific discussion and interaction through scientific advice and marketing authorization and following on. And then the instrument of the conditional marketing authorization, which is when there is preliminary clinical evidence that already allows to conclude that the benefit risk is positive for such medicines that address an unmet medical need and where it is relevant that the patients already have access to this medicine before further and complete data are being generated. And these 3 tools were what our executive director offered to the special committee as the fundamental tools. At the same meeting, she stressed also on the right-hand side that we are still due to further optimize our environment. We need to optimize the research efforts. More patients need to have access to clinical trials, and we need to facilitate their participation. We also need to improve the clinical trials, making them more interesting, bringing the trials closer to the patients, And this involves also to enlarge the community of clinical researchers and the trial readiness of clinical sites. And last but not least, there is the recognition that the clinical development does not stop with approval or reimbursement. And in fact, our perspective, of course, is that the evidence being generated informs the discussions of patients with their treating physicians about what is a safe and effective option that could be considered for the specific situation in which the patient is. So, since these are the main tools that we are highlighting right now, let me on the next slide, please, also highlight you what do we have as collaborative tools available. So these are not product-specific tools, but are collaborating tools that can be considered and need to be considered in particular for rare diseases and rare cancers. These come from collaborations in consortia and for partnering in particular. And what is available are tools, are recommendations, but also the experts and their expertise having contributed to the consortia is available. Let me first mention the innovative patient-centric clinical trial platforms consortium. They finished a year ago or less than a year ago and have produced a number of blueprints and templates, including with guidelines on platform trials. And this is absolutely important to consider because regulators, pharmaceutical companies, academia, and patients work together to create these templates and the framework that can be used. It's readily on their website and we can have a start there. The others have similar provisions, but I'm not going to detail them as much. There is the infrastructure for translational medicine, the partnership, the rare diseases partnership just starting now. There are repurposing consortia that are running now that are able to absorb also medicines that the sarcoma community is interested in and provide support. And I'm very happy that also some of these temporarily funded consortia have now sustained structures that deliver big data analysis or that provide preclinical testing, for example, as a sustained structure following these multi-stakeholder engagements. So next slide, please. There's a number of good news in these legal provisions that may exist since decades, but still are the pillars that I mentioned that can be leveraged with the collaborative tools. And now on the next slide, I want to go into what was new last year and what is new this year. Last year, we saw that the most important, in my view, provision for clinical trials in rare diseases and in cancer and in difficult to research situations has now led to activities at the level of the International Council for Harmonization. And I refer to the adaptive clinical trials technical document, which has been discussed, which is based partly on the reflection paper that we have since long in the EU on aspects that confirm confirmatory trials with an adaptive design. And adaptive designs were elements that are of interest, how to respond to emerging data and how to go about planning trials that are reactive to in upcoming information. And I really recommend to follow what is happening there in order to use it for the purposes of developing medicines for patients with a sarcoma. You see also some other guidances that have been developed in the last year, and there are really enormous efforts to improve the regulatory environment, and I posit them here to be used also for rare sarcomas. Next, please. This year, we anticipate that our initiative to accelerate clinical trials in the European Union will be particularly important and useful for the community here. There will be support for non-commercial sponsors, and this will provide particular support for setting up clinical trials that run in more than one member state, so international trials in the European Union. We also have started a multi-stakeholder platform. An advisory group is at the moment being convened, and this will help to target activities to improve the clinical trial environment and facilitate clinical trials. We will start in January with a workshop on data analytics, such as finding out what were these trials, what were the results, which data sources could be used to inform further trials, maybe generating external control arms, etc. So these are activities that really need to be considered. In my area of work at the agency, in the regulatory science and academia workstream, we will be expanding our offers for briefing meeting with academia as researchers and developers. And we are hoping also to be able to support some of you when then discussions need to go to the products. Lastly, I think the version 6, Pierre, will be released of the anti-cancer guideline this year. And of course, this is the key reference for supporting efficient cancer medicine development. Next slide, please. So let's get now down and get very practical and sit down and take out pens and paper. Next slide, please. How we want to interact and exchange. And here, this is all in one slide, and therefore there's not much detail. But we have a number of mature and proven opportunities for exchange. And I want to first briefly talk about the left-hand side, then the right-hand side. On the left-hand side, it's again about collaborative projects that are in the pre-competitive space, where both the European Medicines Agency and also experts from national competent authorities can be invited to participate in such externally funded consortia. We have already the history of having been in 50 such consortia, and we hope that with the Horizon Europe funding calls and other transnational funding calls, the consortia are interested to invite regulators to be part of these consortia. Now, these consortia work in the pre-competitive space, meaning they do not develop products, but they develop tools. They can look into developing novel endpoints from imaging to patient-reported outcomes, etc. And they have the advantage that they are primarily or already multi-stakeholder. We already have the Health Professional Working Party, where we have ESMO, EHA, and the European Society for Radiology as members, and therefore this is also an entry point for having certain discussions on general principles such as introduction in the healthcare system. Now, on the right-hand side, this is EMA's core business offering for product development. We have early points of contact, and then we have orphan designation, PRIME, what I mentioned, scientific advice. And the scientific advice at EMA is currently complemented also by national scientific advice, in particular for clinical trials. Let me briefly go through it. We offer briefing meetings with academia where they are. No upfront information except for starting points, questions, but no briefing document and no elaborate request form is needed. If academic researchers and developers want to get orientation from the EMA and understand what are the options they consider for taking next steps. We have then as a next step our innovation task force briefing meetings, which look at specific development proposals and convene experts also from the network in order to provide input and have an open-minded discussion about what could be done to support the development of medicines, of drug development tools, of data sets, etc. For specific medicines is the orphan medicine designation. It's not for classes of medicines. It would need to be for specific medicines. And it comes with certain requirements. And with EATRIS, we have produced a training video for all of those who are interested to leverage this procedure in order to have access to incentives. Orphan medicine designation will provide financial incentives, for example, for scientific advice. The PRIME designation I already mentioned, there are certain criteria. We have reviewed this, and we would welcome to have more medicines being either developed by academic researchers or being also introduced by collaborative groups into the PRIME scheme. And then the scientific advice has three elements that I want to highlight to you. Firstly, of course, it can be requested for any aspect of medicine development, typically on quality, on non-clinical development, but in particular also on the clinical development. And any question there, from dose finding to endpoints, that should be chosen, patient target population, you name it. Essentially, it is about all the elements that are relevant for demonstrating that a medicine is safe and effective. We also have broad scientific advice where some of the questions the researchers have are not just for a specific medicine, but would concern a range. And you could maybe argue that some questions in the sarcoma would fall into this category, and we can discuss it then as a broad scientific advice. A third and highly important element is that advice and qualification can also be given on novel endpoints, on drug development tools, on building cohorts that would have a specific purpose as, for example, an external control, statistical analysis methods. All these drug development tools can be qualified, and you will find on our website a list of, I think now, some 30 opinions and around 80 letters of support for such drug development tools. When I last looked the other day, I think there are very few only for oncology. There are some. I haven't found a qualified imaging method for oncology at the moment. So this is an avenue that in particular for topics that concern rare sarcomas is also relevant. The product development and interaction and the tool interaction. So I want to finish now on the next slide with how we want to support you and the development of medicines for patients with a rare sarcoma, and also a little bit what we cannot do. So we want to be engaged on projects, and we want also to be engaged on projects that develop the tools so that we can advance the framework, have new endpoints, and cover this. We want to discuss and offer to discuss the regulatory pathways for specific products, and we are always responsive if you have questions. We respond to phone calls and emails. We still do phone calls, yes. And we have a track record of convening stakeholders on specific issues. What comes to my mind is the Chordoma meeting, the Trizenox meeting for first-line AML, and we also had meetings with stakeholders on specific brain tumors. And I know there have been many more, but we discussed these diseases, and we are ready, I think, to look into how can we convene you together again for some more. We provide the scientific advice, and even though we provide it to developers in confidentiality, please share it. This is meant to inform the community, and it is useful for many to know about the views of the Scientific Advice Working Party and the CHMP. So this can be shared and is important for you to take into account. And occasionally we will also be proactive and try to address public health needs that are urgent. What we cannot do is lower standards. I will elaborate this in just a moment. We think that it's also difficult to discuss general issues only for oncology. And since we have the many links that I have explained, I want to give you the picture that you're not alone with rare sarcoma. We can find ways to discuss it also in a bigger context and make it more impactful. We cannot offer full consultancy, not broker collaborations, and we are relying on the whole participants in the multi-stakeholder development to professionalize and involve relevant parties. And we also have no financing. Other agencies can finance certain research. Next and last slide, please, this one. I want to just reassure you when we say we cannot lower standards. What we're talking of standards, and this is again my personal view you're listened to, is that we are hoping for high quality of the evidence. And it's the quality that I'm stressing. You don't see the word quantity on this slide. The quality of evidence is supported by elements such as the evidence and the tools are fit for a specific purpose. So we call them appropriate clinical and statistical methods. And appropriate clinical methods will include appropriate clinical endpoints. What works in the clinical care? And Pierre, you already mentioned the gap between clinical care and development. And here I think defining appropriate clinical methods starts with thinking how we can bring these elements, care and research, together. Of course, the standard includes that there are minimized other explanations that would explain the therapeutic effects that are being proposed. And I think even in rare diseases, we would like the evidence to show that efforts have been made. And the last element is we need to be aware that when regulators are scrutinizing the evidence, we also are going to inspect it. How were registries created and how was real world data created and how were patients selected for synthetic control arms? So thank you very much for having let me go through this. My conclusion for today is that I think we have already most of the elements of a framework, and what we would like to progress to is doing the casework, looking at real cases and finding ways to discuss the cases, both, as I said, in advice and early interactions, and maybe for developing endpoints in collaborative fashion. So this is for us important to work together to leverage our collaboration to advance the system. We recommend planning for quality, and this includes creating a common understanding of the value of certain endpoints and the meaningfulness across the stakeholders. It's not just us regulators. Please consider HTA colleagues, guideline writing colleagues, etc. And because there is a scarcity of novel drug development tools, my last suggestion is for today also to consider developing these tools together. So it's not just for one trial in one medicine, but we advance the system and are in a better situation in a couple of years. Thank you very much.
Thanks, Ralph.
Of course, thanking the EMA and the FDA, but actually everyone who is participating and listening from outside this room for giving their time today to discuss what we think are really important issues. We also want to be clear that while both of us come from disease-specific advocacy groups, we are here today to represent all patients with ultra-rare sarcoma. And when I say all, I mean all, because the reality is that with diseases that are as rare as these, we have to engage with the global community if we want to really make change. Excuse me. In the case of EHE, for example, today, we have direct contact with EHE patients from over 85 countries in the world. Next slide, please. Josh and I are delighted to say that we have no conflicts of interest. We also wanted to make the point that in the open and honest comments we're going to make today, it is not our intent to criticize any individual or any group. And if we do say anything that appears critical, please believe it, that is not our intent. Next slide, please. Okay, let's talk about patient perspectives. And I always like to start this with a very simple one. Ultra-rare sarcomas are not rare to those who have them. They are very real and they are very frightening. Those patients, therefore, do expect that their disease should get the same focus as more common cancers. Maybe not the same resources, but they do believe that people should be asking the same questions and that the cancer collaboration should be working and fighting their corner for them. They also passionately believe that the lower commercial potential associated with these ultra-rare sarcomas is not an acceptable reason for lack of progress. Put simply, their lives are not of lower value. As we are here today with the regulators, it's worth just mentioning the view for patients of the regulators, and that view is very simple. They believe the regulators, or they hope, are working very hard to get safe and efficacious drugs approved and to them as fast as possible. In terms of expectations, these are fairly simple. They want everyone, all of us in this room, in the cancer collaboration and those outside, working hard together to find better treatments for them. They don't want the rarity of their disease to be used as an excuse or a barrier to progress. And they hope that we will also work hard to find appropriate procedures and not fail to make progress, because procedures developed from common diseases don't seem to be appropriate. Ultimately, in its most simple form, what they hope is if they don't survive the disease, it's not because we didn't try bloody hard to find solutions. We also wanted to make the point that the patient communities of these rare diseases are hugely motivated. They can achieve an unbelievable amount, and it's humbling watching what they do achieve at times. They're there to help. Mountains can and will be moved. Next slide, please. In terms of challenges, of course, I don't need to tell people in this room that for ultra-rare sarcomas, practically every aspect of the cancer process is a challenge. And that is largely due to the fact that patient numbers are extremely low in any area. And that has a knock-on effect, unfortunately, such as reducing the political and commercial drivers that drive the process for more common diseases. Today, of course, we're talking about the issue of finding and approving new drugs. And so we particularly wanted to focus and mention the paucity of effective treatments for these diseases. This is again largely due to the lack of patient numbers and the difficulty in collecting meaningful data sets, which means that often the understanding of the biology of the disease is at a lower level than for common diseases. That is why we have to look globally. And I think, Pierre, yesterday you mentioned the need not to waste resource and find every way that we can to maximize the information we have. Sadly, most rare sarcomas do not have disease-specific treatments approved for them. And the treatments that they may access through general categorizations, such as soft tissue sarcoma, don't work. So I cannot overstate the importance of looking for, finding, and approving new treatments. But sadly, it is a fact that with such small numbers, for drug development companies, they have a far lower tolerance for risk, for uncertainty, and for cost. And a result of that is that repurposing and repositioning of existing drugs will always be a critical pathway for finding new treatments for these diseases. Okay, those two slides have been some very general points, but what Josh and I thought might be useful was to also make some observations about actual experiences in a little bit more detail to try and give them a little bit more focus. So I'm now going to hand over to Josh for the next couple of slides.
Thank you, Hugh. As you mentioned, my name is Josh Sommer. I'm co-founder and executive director of the Chordoma Foundation. I'm here as a two-time chordoma survivor, but also representing a global chordoma patient community of thousands of patients across the world whose lives and futures really hang in the balance and stand to benefit from the work that is going on in this room today and from the clarity that I think we all hope to be able to generate through our conversations together. I think more than anything, I just want to say thank you to all of you for the attention that you're bringing to this important topic. It really is truly personally meaningful and meaningful to the many thousands of patients across the world who are in urgent need of better treatments. I think to Hugh's point, there was a lot of specifics that we could go into about chordoma and these other rare cancers. However, I think from Ralph's talk, which was really music to my ears, I think there is a recognition and an understanding of the challenges that these diseases face in terms of clinical development. It is so heartening and so encouraging to hear the recognition of that and to hear that you have a framework in place. So I love the idea of now is the time for the casework. And I think that we as patients and as the Chordoma Foundation, and I think as many patient organizations, stand ready to do that casework with you. I think one thing that's just worth underscoring is that in the time, in the 17 years that I've been involved in the Chordoma Foundation, and that many of us in this room collectively have been working to drive forward chordoma research and treatment development, a tremendous amount has been learned about the biology of the disease, which is now pointing to very promising potential therapeutics, both based on preclinical data as well as early clinical data. The conundrum that we face, though, is that because of the absence of clarity with respect to clinical development paths, with respect to how drugs for chordoma can get approved, this is creating an impediment to designing clinical trials, both for investigators, but importantly, it's creating an impediment for companies to invest at all stages of drug development, whether that be repurposing, repositioning, or new drug discovery. So I think more than anything, my hope is that we can generate clarity on appropriate endpoints, on appropriate next slide, please, on appropriate external controls or historical controls, so that we can unlock the latent energy, the latent potential, the latent investment that exists from physicians, from industry, from patient organizations to move forward clinical development and ultimately the discovery of effective treatments for this disease. So I think a key question is whether their guidance and whether that clarity can be generated for groups of ultra rare sarcomas, or whether it needs to be generated on a disease by disease basis. But I think just to underscore, we as the patient community and as the Chordoma Foundation and representing many similar patient organizations stand ready, willing, and able to help in any way possible to achieve that clarity, and Ralph, to do the casework that you described. So huge thanks to all of you in this room, to the EMA, to EORTC, to the clinicians here for the intellectual effort and input that you are providing and the energy that you are bringing to this important cause. We really greatly appreciate it.
Thank you, Josh. Could we have the next slide, please? What I wanted to do on this slide was just highlight in terms of opportunities that some of the principles we need or think about are already in place. And this is a summary slide of the EMA's own micro, small and medium-sized enterprises guidelines, or SME as it's called. And when we go through this, what we find is that the objectives, the role of the SME office, the assistance it provides, the ongoing dialogue that it facilitates, and the reduction in fees, for example, that are available. These are all attributes that would be hugely useful to an academic patient-led consortium. The only issue with this at the moment is it appears to be only available to companies, which means that an academic patient-led group couldn't access these. Or maybe that's our interpretation, and maybe we can, and that's certainly something we'd like to understand, because if we can access these sort of benefits or help, then it will go a long way to helping us move forward, and it will potentially ensure that profitability is not a barrier to progress. Next slide, please. What we also thought would be useful was just to look at what the process we went through and are involved with the EMA through the pilot scheme that was introduced by the EMA to try and help with repurposing for drugs for rare diseases. On the left-hand side of this slide in the red box is that experience. And actually, we made good progress. We progressed the scientific advice process. We gained an understanding of exceptional circumstances. We understood the potential necessity for hybrid applications, the potential benefits of orphan drug status, and indeed the potential need to introduce or involve the marketing authorization holder later in the process. So that was excellent. The only observation I might make is that did take us a year to achieve. And part of the barrier, I think, looking back, was that much of the communication was by email in the form of questions and answers. And that actually, with hindsight, is quite limiting in terms of creating an environment for open discussion and brainstorming, and is slightly slow, or is slow and a bit sterile. And it certainly didn't give us access to those SME benefits. But that is not a criticism of the process. That is just reflecting perhaps our lack of understanding and not asking the right questions. What we did on the right of this slide in the green box was just think about what might an alternative process that might work. And this is just one set of ideas in which we would start again with the application being submitted by a group, a dossier for a drug that they feel needs, and they have evidence to justify a repurposing and approval for label extension. That could lead to the EMA, if they agree with that, then actioning a stage 2 process, which would be parties coming together in a working environment over a 1 or 2 days, maybe over weeks, but ultimately seeking to try and agree preliminarily, at least, on key issues like the validity and presentation of existing data, an appropriate application pathway such as exceptional circumstances, the design and magnitude of new data that could be required, and access to the benefits of the SME process, which we saw on the previous slide. And if those preliminary agreements are reached, then moving into a third stage, which would be basically working to develop an appropriate marketing application, and at some point engaging with the marketing authorization holder, but potentially at a point of time when we have greater clarity of process and costs, which is important. Next slide, please. So moving forward, in conclusion, we're very encouraged because we recognize there is a growing awareness and desire from many groups, including, as we've seen today, from the EMA, to try and help ultra-rare sarcomas. The EMA's pilot scheme was an example of this, and the proposed legislative changes, Article 48 in the EU, is another example that is testament to the desire to make a difference. So for patients, the direction of travel is very good. The speed of change, though, is potentially for patients slow. So I want to finish with a challenge, a question, but really a challenge to everyone in this room and to those outside it, on behalf of all ultra-rare sarcoma patients. And the challenge is, can we establish the open and ongoing dialogue that will enable us to find the solutions we need, the answers to the key questions, and that ultimately will deliver what I think is a common goal for all of us, and that is to deliver new treatments that work to ultra-rare sarcoma patients everywhere. Thank you.
Thanks a lot indeed. And I would say that this presentation was almost a summary of our workshop. You asked the right questions. It's very practical and very realistic. My reaction would be that you're asking the right questions, and you underline that at the level of the regulatory agencies, we have already all the tools that could help, but they're not grouped in the right box. We have to take a tool in one box, another tool in another box to answer all the questions. And for stupid rigidity questions, this is not always feasible. The idea is to break the boxes and let's build on positive experiences. I'm just taking 2 minutes we will discuss the repurposing process with EHE. I had the honor and the pleasure of being a coordinator for that procedure from the side of the EMA. We could, just because we tried to discuss on this specific question, we tried a process which was not the right one for the discussion. We decided to break the process and to engage into discussions. And I believe this is exactly the model we should follow. So it's a pity that if we waste data, if we waste resources, if we waste patience, if we waste processes, this is a lost opportunity for anyone in the process, for the patients, for the academics, for industry as well, but for the regulators. So let's stop being stuck with rigid approaches. And I just told you that at the level of the Scientific Advisory Working Party, as an example, we are trying to reflect on the fact that we could make better use of the discussion meetings that are not really the best solution for all the procedures. Just to give you an example. So this is exactly the type of discussions we should have today. Thanks a lot for having been so clear and so practical. Really appreciate it. The next speech is by Denis Lacombe. Thanks, Denis, from the EORTC, on the major challenges and opportunities, complementary to the speech we just had, I believe. Thanks, Denis.
Okay, so thank you, Mr. Chair, for the introduction. Yes, my name is Denis Lacombe, and I'm the CEO of the EORTC, and I was asked this morning to share with you our, I would say, perception about challenges and opportunities. Can I have the next one? So I have no conflict, the next one. So I think I should start by a disclaimer, and because so that you follow what I'm going to say, I think I am a proponent of randomized clinical trials, and randomization does not necessarily mean a comparative phase 3. I think it can happen on very small number of patients. It can happen in real life, and it can be extremely pragmatic. And I would say a little bit, if you allow me to say so, and I know I'm talking to a community of ultra-rare, dealing with ultra-rare patients, but the rarer, the more we should try to randomize, because these patients are so rare that we should put them in the best possible design clinical trial. And again, I think there is no reason why we should not offer the same level of certainty to rare cancer patients. And I guess it echoes a little bit what Ralph has told us, no lower standard, and demonstrate that we have done the maximum effort. So I think single-arm trial should not be the default position, but the fallback position first. Can we do as much as possible? Of course, the design that we ultimately select should depend on the question, but of course, something that is critical is the understanding of the biology. And having said so, whatever design we come up, I think we should have a clear understanding of what we can claim about it. Can I have the next one? So to share a little bit the challenges we've been through for these trials, rare cancers, ultra rare, maybe sometimes not so rare, but not so rare, is of course access to patient recruitment, epidemiology data, where they are, how many really are they, the natural history. Challenges, of course, to stimulate the commercial sector to work with us, also to stimulate sometimes independent funders. Often because they can be longer trials, so it's sometimes difficult for charities to commit. We have to admit also that in the academic sector we are fragmented. Maybe we should work on having more efficient collaboration. And certainly regulations. Regulation have been, for us, a limiting factor in their pragmatism to activate clinical trial, especially when you have to do it, for instance, internationally or intercontinentally. So that's something we should look into it. And of course, methodology. I will not come back. I think I've said what I wanted to say on the previous slide. And of course, what is important also, the challenges is how to have optimal translational research programs so that we can understand the biology. Can I have the next one, please? So having said that, I think it's best to share our experience based on examples. So I would like to share this one, which is a trial which was conducted at the EORTC some years ago, addressing relatively rare entities which are listed there. So it was a basket trial. It was a classical phase 2 based on a master protocol and different arms. What I just want to say on this example is that if I took an example, the IMT example, the inflammatory myofibroblastic tumor arm, is that we put 55 patients, it took 55 months to accrue 35 patients in 30 sites. So the question is, could we do better? Because I think we have the expertise to do basket trial. We have the expertise to do all these designs. The problem is, do we have the optimal solution to operate this trial basically in the healthcare systems? But again, the expertise is there. Can I have the next one? I want to show you another example, and here we are switching gear. It's an observational platform which was actually communicated at ESMO last year, and I think it's quite an achievement, because basically it's a prospective collection of almost 1,000 rare cancer patients across 10 different type of organs. So it was quite a large platform. And what is interesting is that we could do that in 3.5 years in 14 countries. And we had, for the molecular profile, a turnaround time of about 14 days, which is also an achievement. But the reason why I'm saying that is because there are solutions. And what we are trying to do here is, what can we do better? And what I am particularly interested is that we could identify actually for more than 600 patients relevant molecular alteration. And in the last point is that quite a substantial number of patients were offered some recommendation to reach a clinical trial or to get access to a drug. The limit, of course, of this program is that it's an observational platform. Fortunately, we did it on the backbone of our registry, SPECTA. So through the SPECTA platform, we are now following up these patients for what type of treatment they have received, whether it was changed based on the recommendation, and what are their outcome. Ideally, you would have seen, of course, this as an interventional program with, of course, bringing drugs to these patients. But I can tell you, doing such a large interventional international program in Europe today is quite challenging. But again, the expertise is there for the future. So it's how can we optimize that and build from that. Can I have the next one, which I am a little bit humble to present because the study leaders are in the room. So it's I would say a more classical phase 3, indeed not such an ultra rare type of disease. But what I want to point out is that we have opened Strexit, which is actually an observational arm next to the randomized clinical trial for those patients who are not necessarily eligible for the randomized clinical trial. What I want to highlight here, because you will see when I come to the wrap up, is that it is an intercontinental trial. So when it comes to difficult, challenging clinical situation, again, it's impossible to do things alone. And here there is the expertise, the capacity to do intercontinental trial. And additionally, of course, what can be relevant for patients is that it has a quality of life dimension, because we have, of course, this expertise at EORTC as well. Can I have the next one? So what do we need optimally? And if you can go through the slide in the interest of time, I try to group a little bit in 3 different categories based on all this expertise, and I would say successes and sometimes failure that we have had, is what, in terms of feasibility and assessment, I believe you've seen with the basket trial, it may not be so efficient. So I think we need some registries on which we could plug these trials. And this registry could be a solution to access to the natural history and giving this information about the longitudinal follow-up, what we are trying to do now for the SPECTA, for the Arcagen patient in the SPECTA observational trial. I think it could help us raising hypotheses to do clinical trial. And again, coming back to my statement about randomization, I think there are new approaches like tweaks, trial within cohort, that can be quite interesting, possibly to be tested. This is the extra mile that we should be doing. I think they would help us, of course, if we would be working from registry to benchmark the intervention. And of course, we would have to see how we can optimize the recruitment, possibly through a limited number of institutions because of the complexity of interventional trial. The second type of challenges that I try to group is data access. I think you've seen what we've been trying to do in terms of access to genomic solution, to molecular alteration. Also, the capacity that we should develop is having access to reliable external control. And indeed, despite the will to randomize, we have to admit that there are situations where maybe it's not going to be possible. So having a registry could be, of course, a source of extremely high-level quality data to have the synthetic control arm, and of course, to do translational research, ensure the quality of the data, because we know that if we don't have some control, garbage in, garbage out is still very valid. And I think also we could do better in supporting data sharing exercise. And the last, I would say, group of need that I have identified are partnerships. And that's why I have identified also the STRAS2 Strexit program, is the fact that we should look at global programs, also outside the EU partnership. I know that later today we will have a presentation from the NCI, and I was very interested already to hear the MyPart program in the US, which bears a lot of, I would say, resemblance with what I've described to you. I think indeed optimize the interaction with the commercial sector. And last but not least, what we are doing today, discussing with you, the regulator, but also indeed the downstream payer, the downstream stakeholder, such as the HTA body and the payer, those who give access. But maybe we need to deliver, I would say, better type of data set, at least demonstrate, make all the effort that we can do so. Can I have the next one, which I believe, so the opportunities for me is there is a lot of experience around how can we get the best of the two worlds. You've seen what we can do in an observational setting. You've seen what we can do in an interventional setting, and how can we bring that together actually. And this is my last slide. So I think we need to optimize existing solution before we reinvent the wheel. So I think we need to re-engineer partnerships with existing infrastructure. Again, I believe in registry with interventional trial, the combination again of the two worlds. I think Arcagen would not have been possible by EORTC alone and would not have been possible by Huracan alone. So there are existing infrastructure that are very efficient, that have complementary expertise. How can we optimize that? I think we should redesign regulatory solution. I cannot enter into the detail, but certainly we have obstacle because this type of sometimes different trials are not necessarily easy to process. And again, can we give a look at other approaches like tweaks, for instance, what should be the optimal external controls, decentralized clinical trial. And again, I think we can all do that, of course, if we have co-creation and partnership, and that's what Sylvia has been advocating for in her introduction. And I think this is my last slide. Yes, I was asked to identify possible relevant questions. So basically, how could we create an ecosystem for rare cancer where actually the efficiency, the result can be better than the sum of the parts that we have today? What are the optimal data sets in terms of design and the endpoint, which can be suitable not only for approval, but also for access. And I think that's what Ralph has told us. And again, because I still believe that we will have to do a bit of this work without the commercial sector, is how we can structure the work of academic research in regulatory decision making, and of course, stimulate access to relevant new agents. And with this, I would like to end my presentation, and thank you for your attention.
Thanks a lot, Denis. Very, very focused and really interesting. I was not expecting less. One message which is important to me, but I could be wrong, is the fact that we should not focus only on the clinical trials. This is very important. But besides and before the clinical trials, it's very important to collect data. And you nicely demonstrated that longitudinal collection of data, even outside the clinical trial, could be very important. Because we will group as an example, the prognostic factors, the natural history of a disease. If we are careful in collecting samples, we may discover that even biomarker we do not know at the time of the collection, then retrospectively could be a fair indicator of the prognosis, of the sensitivity to a given treatment. Could be a marker for a new subgroup, even in a rare disease. Everything is important, even the quality of life, what does matter to the patients, as an example. And questions outside the trial of a given drug are massively important. This to say that all data that could be collected, not only when trying a drug, but even from the simple, not that simple, observation is crucial. We shouldn't lose any data. So this is the message, and we should organize ourselves to group the observations, to build networks, to work together, and to help in understanding what we have already available. This is very important. Thanks a lot, Denis. It was very clear. The next speech will be by Kate Roos, who is the chair of the Methodology Working Party. Thanks, Kate. So I trust you will be sticking to the timelines.
I will do my utmost.
I knew that. Thanks a lot.
Yeah, well, thanks a lot for the introduction. And to add to that, I'm not only chair of the methodology working party, but I'm also working in an academic medical center. So a part of my brain actually also assists developing or repurposing drugs, for instance, for the rare salivary gland cancer. So I kind of very much find the dilemma of on the one hand, let's say retaining the high regulatory standards versus making progress for actual physicians and actual patients. It's a kind of everyday dilemma. Moving on, next slide, please. What I will do briefly is also clarify the single-arm trial reflection paper, because it came up already. It's been driven, but not covering only rare cancers. And I think it's good to also position it well, because I think the most important point I would like to make is the third one, is that we need to think very broadly on the design of clinical research for rare diseases in general, but certainly for rare or ultra rare cancers. And I have a couple of suggestions how we can facilitate that in the way we interact regulatory. Randomization was already mentioned by Denise as being very important. I think we do that sometimes a little bit too easily, putting it at the top of the hierarchy of evidence, because also that very much depends on the question and the research question you want to address. And also there are limitations to randomized trials because you create an artificial situation, which may actually not necessarily be the optimal evidence for every research question. So next slide, please. So I'm very much, in that sense, very much in my mind, I come from an experimental design area. There's a research question, you have a design, you get results, and you go back and you answer the research question. And I think the controlled experiment, the control part of a randomized trial, is not only the fact that you have a randomized control group, it's also that you have very good control on everything else that happens in the trial. And that standardization of assessments, timing, following up patients. And I think what we see and what we luckily see in collecting registry data is that we try to get as close to that part of a controlled trial as possible. And I think that's hugely important to increase the value of registry data. So that's one point. The other point that's usually not emphasized, but quite important, it also allows you a good, well-controlled randomized trial to assess the uncertainty that's left with quantitatively very well. That's much more difficult in observational research. So we might actually be underestimating uncertainties for other designs. So that's one of my methodological challenges. Moving on to the next slide, please. The single-arm trial reflection paper. I'm not going to deal with the whole paper, but I do want to clarify what our approach has been. So we have had actually lots of discussions, particularly on this reflection paper in the context of rare cancers, because we see them happening there. We see quite a lot of approvals based on single-arm trials. So we wanted to get our heads around on how do we look at single-arm trials more generally. What we did do is limit the scope. What we did not do is cover all the designs where you include an external control in whatever way to answer the primary research question. And I'm quite happy we did not, because that design space is large. And the last thing we would want to do was actually limit that design space with too much guidance. So we focused on single-arm trials that can stand on their own. You've submitted the protocol to an ethics committee. There the design and the research question is described. What do we think actually is necessary as part of such a design to be able to interpret the results pretty much as a self-standing trial without immediate and full integration of external data? Because I think there the design space is much larger and we should give that careful thought. Next slide, please. And the key point, I will not cover everything, but one key point is the concept of if that's the vision you have on a single-arm trial, you need an endpoint and a context and disease understanding that will allow you to isolate the treatment effect. And that can actually be quite challenging. And I think it's actually challenging for some of the ultra-rare sarcomas. And underlying the discussion is objective response rates for RECIST criteria might be suitable to isolate the treatment effect, but it might not be very relevant for patients. So this concept is there. This is the background to be able to isolate the treatment effect. But there is the underlying unmet challenge there is, are we capable in a particular context to come up with the endpoints and the setting that we are able to interpret a single arm trial. But my point will be, if that's not possible, you might need a different design. Next slide, please. And finally, also, but I can do that briefly, since you're in an uncontrolled setting, all kinds of things that we think are generally important become more important. Pre-specification, not interfering with the conduct of the trial, being actually in terms of execution of high quality. All of that, if it's not there, increases uncertainty. You're actually quite unable to assess. So single arm trials are maybe not even that easy to execute. Next slide, please. So that's giving some background here, but my point is more mainly, we should probably think more widely about exactly what type of design we are looking at. So my point is widening the design space. And for your reference, there are two papers there. One is on the use of external controls, which is actually collaboration between industry statisticians and regulatory statisticians. So there is a kind of shared view there. And the other paper I would like to highlight is on glioblastoma, where essentially the same, you will see some of the elements I will be presenting here as well, but then specifically for glioblastoma. Look differently at designs. And these are three areas of design, if you like. And the blue parts, and that was emphasized by Pierre, you might look at those blue parts as well-controlled collected registry data, either retrospectively or prospectively. So you have, I think that's part of the crucial thinking here. You have external control data, which is actually pretty good. And then going from top to bottom, you have non-randomized control, but you plan it prospectively, which doesn't mean that the data needs to be there, or prospectively, that can be retrospectively as well. And you have different shapes and forms of including randomization, which I think is still quite relevant. So the second one is you have randomization, but skewed. You limit the control group randomization and you augment your data with matched controls from the well-controlled registry data. And the square there indicates you could actually do an interim analysis to decide whether that's achievable and how large these groups have to be. So there is some adaptive element there. So that's an option. Another one, briefly mentioned by Denise as well, is the TWIX design, where you have a prospective cohort of patients with comprehensive data collection. So you collect a lot more data than you would do in usual care, and you do it up to the standards that you define. And instead of randomization, you actually, at points in time that a new treatment option presents itself, you randomly select from that cohort the patients that will get the treatment, and the ones that are eligible but you did not randomly pick are your control patients. And you can do that with one intervention, but over time you can do that for multiple interventions. So it's an attractive design, and there are certainly in the Netherlands there have been a couple of trials, at least started and a few completed. So we've summarized both the experience here as well as methodological reflection, because usually more complicated designs do come at a price. So they are slightly more complicated, but they are a way of introducing randomization, which has proven to be easier to implement. Next, please. And as such, general statement on platform trials. I think we share the vision, whether it's regulatory or clinicians or patients, that they provide a route to make it more efficient, quicker to develop. So full support there. It may even provide also there opportunity for randomization in challenging issues, because most of the patients will actually get active treatment. And while preparing guidance is there, we do see that we need some fundamental thinking there, because our idea on what constitutes an experiment, issues like type 1 error control, I think we need to go back to the drawing table to fully understand how that plays out in platform trials, because we run the risk, from a personal statement, that if we are too strict there or too dogmatic, we might actually not reap the advantages. And I think we should. Next, please. I want to go through one example, different disease, but it's also lethal. People with this diagnosis die within typically 3 years, but can be quicker or up to 5 or longer. There's no real treatment that helps every patient gets riluzole because there's some evidence of prolonging life for a few months, but you could also interpret that as prolonging suffering for a few months. It's about 500 patients per year, and there's a very good registry, both in the Netherlands as well as European-wide. Next slide, please. And what we did there is, I hope the next slide will come. What we did there is see, is a hybrid design which I showed, augmenting a clinical trial control data with data from the registry. Does that actually give you benefits? Is that more efficient? Is it quicker? And a trial was performed, repurposing lithium carbonate for the treatment of ALS. Very quickly, straightforward, randomized, placebo-controlled, blinded trial with a primary endpoint of survival. And it was a sequential design, so multiple looks and enabling the trial to stop early for futility or efficacy. And in this case, it was stopped when 61 of the originally planned 133 patients reached the primary endpoint. Next slide, please. And what we did is actually look at this trial, and what if instead of doing this trial, we've had augmented it with control data from the registry in which it took place. And as I mentioned, there's a good registry. Next slide, please. And here we can actually, and that's the ability you have with a good registry, you can truly match. You can look at which patients would have been eligible from the registry, but we went even more detailed, which were eligible and could have actually entered the trial because it's in the same time frame. So from start study between stop study. And if you do that, you come from 340 eligible patients to next slide. 133 matched controls that could have entered the trial, propensity score matching underlying this with an equivalence criterion. And in this case, you could actually also check on the survival distribution. Does survival in this matched control actually mimic the survival we've seen in the clinical trial? And the top one is the large group of matched controls. The second one is the fully matched controls. And you see survival curves matching. And what would be the impact of the trial? That's next slide, please. Very briefly, this was a sequential design. What is evident from adding this augmentation from a very good registry is that you could have made the decision to stop earlier. Of course, this is not good news for patients, but it is good news in terms of speeding up development. You stop something that's not successful, in this case, half a year earlier, and are able to offer something that probably has more potential. So that's kind of sketching you a real concrete, tangible benefit of looking at different designs. Next slide, please. Suggestions. I'm afraid I'm in line with Denise. So I do think randomization is really important and more important than the dogmatic powering for 5% two-sided significance. Randomization provides you a very solid scientific interpretation of your data first, and the 5% is secondary. I think it's crucial to have prospective designs that include an adequate control group from the start. So preferably in the protocol, even if the experimental part is only is a single arm. Think about think and include an adequate control group, because that's also going to be relevant if you want to reach patients and have treatments reimbursed. This is a quote from the paper on the external controls. A critical factor will be to maintain the same high level of scientific rigor associated with RCTs. And you see similar phrases very often raised. I think, to be honest, that they're not very informative, because we do not really articulate what we think is scientific rigor if the design is not exactly the RCT. So I think we should distinguish that from classical benchmarks as type 1 error control. We should be more specific on what we consider the scientific rigor that we will want to achieve. Next slide, please. That's my final one. And I think we need a framework there. So to have an intelligent discussion, if we have an innovative, different design, how do we benchmark that? How do we actually assess that that's indeed addressing our needs to have the standard or the scientific valid evidence in this particular setting? Given the diversity of designs, I do think it's not only let's say, I think we have already had good suggestions on changing the interaction at a regulatory level. I think it should be open and early. And the content, in my view, personal view, is the content could be different. I think we should not only share the ambition to actually get something done for patients, but we should also share the ambition and acknowledge that we are scratching borders on scientific rigor that we are able to achieve. So let's start that discussion as early as possible, and also open, be frank, because usually the other side of the table that I know, they see the same problems in terms of the quality of the evidence. Let's be open early and see whether we can jointly come up with the best solution. So I think the content of our discussion could probably beef up to come up with better solutions. And last but not least, re-emphasizing again, invest in prospective registries. And I think that's actually a huge challenge we often ignore. Having sustainable long-term registries in place for rare diseases is really difficult, but if we achieve it, I think it will help lots and lots of clinical trial research forward. So I think that's not sure whether we as regulators should play a role there, but it's something that we should all strive for, because I think it's crucially important. Thank you.
Thanks a lot, and you are very efficient in being short and even very understandable. Something I like when listening to statisticians is that they all underline the idea of the risks and of the scientific high standards. The scientific high standards are something we cannot discuss. It is necessary to have high standards. But whichever the conclusion is associated with the risk that people outside the statistical world sometimes forget. We have a risk of concluding wrongly to something, but that risk must be taken into account respective to the context. Sometimes we are ready to accept a higher risk. The simple example is the 5%. 5%, it is a convention. Sometimes we could accept the risk of being wrong in concluding to superiority, a higher risk, because we believe that the risk of not concluding is higher to the patients. I do not say this is the solution. I quote that as an example. Every development which is not based on two fully powered randomized trials is at higher risk respective to this type of experiments. So just a short intervention, because normally we have the questions at the end, but if you want.
Yeah, forgive me. So thank you, Kit. I really enjoyed that presentation. I have one question that leads on to another question. So your trial in ALS, was that assessed by the regulatory authorities? And the data from the registry, how was the definition of high quality data assessed? What was it deemed acceptable?
Quick answer, no, this was not assessed by regulatory. The registry data is also part from a larger effort where also company-sponsored trials are being done using partly the same registry, same center, same research groups. So it will come under scrutiny.
Yeah, I suppose that's my fundamental question in a way. How do you define high quality data? Because it's all very well talking about real world data and registries, but how is that definition of quality made in order to get regulatory approval?
Quick response, but maybe we should actually flag this as one of the things that we need to follow up very, very well. But it's there. I think there is a bridge to real world data here, and we have a data quality framework guideline out there to at least present our initial thinking on what presents the quality data from observational research. So we are setting steps there, but I think it's a very relevant point to even go beyond there.
Yes, and indeed, we are having in February a workshop on the registries and the collection of the data that could be connected to your question. So it would be interesting to follow that one. We don't have to wait for long. Patrice is organizing that one.
Yes, just to complement that, we will even have a specific chapter on real-world data that is currently under development for this data quality framework, so it may definitely help to answer your question.
Thanks a lot. We are answering a question by a question, but the question will receive an answer. So, Valerie, the floor is yours. A good example on how we would try not to waste data and to take an opportunity to collect interesting things to treat and to investigate the diseases.
Thank you for giving me the opportunity to present our special authorization programs in France. So I am Valérie Denux. I am a physician also by training, and I am the director of Europe and Innovation of the French Regulatory Agency. So our program are focused on helping, to solve some difficult medical situation for a really specific patient, but also focus on accelerating access to the treatment for patients, especially innovative treatment. So it's really well used for a rare disease, for example, an oncology, as I am going to show you. So next slide, please. So just to show you that we elaborate these different programs all along the process to develop a product. So we have a classical clinical trials, and it's really important, and I totally agree. However, we develop some programs I'm going to develop a little bit just after, all along, so before marketing authorization, but also after marketing authorization. So next slide, please. So I'm not going to detail, but just to tell you that we have a first program. This first program is compassionate use, nominative compassionate use. It's case by case, patient by patient. So it's especially well adapted to patients that are rare disease with no solution, or oncology also in a really last treatment, and then a very no solution also. So we have two way of doing with this this program. The first one is, okay, you have some drugs existing in other countries, not in France or through the centralized authorization in Europe, example in United States or other countries, and then we would want to use this this molecule even if we don't have any authorization in France. So it's possible to do that, and it's also possible to use this program with a drugs that is in development process, no marketing authorization anywhere, nowhere, but we have the possibility to use this program also if it's on a development process. It has to be asked by the health professional, that prescriber who said, okay, we would want to use something and to find a solution for these patients. Next slide, please. So just lesson learned here, just to show you that oncology is a main part, because you have 10,000 special nominative authorization per year in 2023, and also you can see that the different part of oncology are really you have a lot of solid oncology, but also hematology, and we identify sarcoma, especially for today, and so you can see that we use this program also for in this case. Just to tell you that when you have this nominative special authorization and the drugs is on the development process, we have to ask to the laboratories to be engaged in after that asking for early access for every patient. So that means that okay, they do the development, and when after quite finishing the development, just before marketing authorization, they can ask to open the access to all the patients. If we agree with that, even before the marketing authorization, it's available in France. And also they can do that after marketing authorization. Why? Because we are quite long in France sometimes to say to agree on the price. So that give the opportunity for the patient to have access to the drugs, even if we didn't agree on the price. So you can see that this year we had more than 900 patients who has been delivered on oncology products in a pediatrics area, for example. And also you have 12 new drugs that are being used like this, and then they have the commitment in the early access for all the patients. Next one. So just to tell you that we have a special portal on that. For that, this is the prescriber that I can access, and pharmacists that can have access to this portal. The good point with this portal is that you have a main part of the authorization that are done automatically. So we delivered in 2022 65,000 authorization in a nominative authorization, and this year we did 60,000, and 90% are automatic. That means that the first time we have the request for one drugs and one indication, we are doing the analysis, and we manually say yes or no, and also we define, is it possible to make it automatic or not, if they respect the criteria, and then we say yes or no on the machine, and then it gives us the opportunity to deliver a part automatically. Next one. So this is the early access I spoke just about before, just to tell you that after this nominative authorization, we want that the main, all the patients that are necessary with this treatment can have access. So we ask after that to do a global authorization. So more than 100,000 patients had this treatment through this special authorization since the new reform in July 2021 on this special authorization. Next one. So you have the lesson on here also, and you can see that the oncology is really important in this area. You have mainly this is the oncology and the hematology in this early access in this program. Next one. Just to show you that we did a study with different countries, and we can realize that focusing on the drugs that enter in these programs, that we can have access to these drugs 11 months before, the first commercialization in the different country in Europe we had for this study. So that means that we have a real acceleration also for us in France, because if we wait for the price, it's going to be longer. But with this program, it's really, really good for the patient, because they have access really early to the new treatment. Next one. So the last program we have is this one, is compassionate prescription program. It's a very interesting program, especially for rare disease, and because in a rare disease area, we have a lot of off-label use, and especially in pediatrics also. So we built this program to say, okay, we need to work on this off-label use. We need to know what they are doing, and then we need to help the health professional to see if really or not these treatments are of benefit for the patient. So next one, please. Oh, next. Yes, complete one. Thank you. So just to show you that this compassionate prescription program is the process is this one. Okay, you have a drug with the marketing authorization in a 1 or 2 or 3 or many indication, but this drug is used on an off-label use outside the indication of the marketing authorization, and then we collect data, but normally it's not a really good collection, with a high quality and everything. So we said we work a lot with the Rare Disease Association, and we said, okay, you need to collect the information on this way and this way, and they have a registry, and they put all the information on the registry, and we can use all this data for a first step to say, yeah, okay, do we have to do this kind of program or not? If the benefits seems to be high or good, and then if yes, we authorize the program for 3 years, and in this program we ask for a collection of data, really focused collection of data within a framework, so specific data, and then we would want to use this data in a clinical trials. But we are working currently with the methodologists, to say, okay, we would want to include this data, but really organize data after the 3 years in a clinical trial to go faster with this clinical trial, to have necessity of less patients in these clinical trials, and then we could do repurposing. We discussed with industry. Industry said we would be interested in that because this is off-label use, though, so that means that you have a market for these drugs, so they are interesting to work with us on that. So let's see, because it's quite new. Next one. So you can see also in this program that you have oncology also, and for the moment you have 28 this compassionate prescription program, and 50% are on rare disease. Currently, we are working on 14, and also we had potentially 91 because the Rare Disease Association provided signals. So we have to work on that and to see what kind of data we have to make it possible. Next one. And conclusion. So thousands of patients, as you can see, access to our special authorization programs. We really have an acceleration to the access to innovative treatment for them, and also we need to collect all the data. So this is really a way of doing, but because this is since 2021, even if before we had this kind of program, it was less structured, but now we need to improve data collection. We need to improve organization with that, and also we need to improve our own way of doing analysis and how to utilize and how to design clinical trials with this data. Thank you. I went faster as I can.
Sorry, may I ask a very quick question? Thank you for this presentation, but how do you define which off-label compassionate treatment you allow? I mean, do you have a commission? On what do you base your decision? Because in my view, I mean, I understand the process, and I would love to have a similar process in my country. But on the other side, I see this as a failure of the European mechanism, because it means that we try to give outside the label what we think is requires a need. So I think it would be interesting, maybe for another workshop, to try to understand at the country level how off-label use, compassionate use is handled, to try to understand which are the criteria where use at the local level to approve, to allow the use of such a drug, and understand if this cannot be also used at a global European level.
Yes, just very quickly to tell you that I would be really happy to have a workshop dedicated to that. I really think that we need to work all together on that. If it is on the program, on nominative programs, this is for our we have medical division who are working on each case. When you have a request for one patient, this is our medical division who is working on this case. So to provide the authorization, and we have a process. I'm not going to describe now, but we have a process for that to guarantee maximum, the quality of the decision. For the early access program, this is a committee, with HTA. We share with HTA the decision. And for the last one, we are working also with we have a scientific, committee, and we also have our medical division. So we have also a process for the decision for this. It's quite long for this CPC because we really have to be sure of the decision.
And I would add that most CPCs are coming from signals made by the learning societies. So this is a security for the program. So it's not something coming from a given physician. It's normally something that a learning society comes and says, "Oh, we have a need for this drug that should be used off-label, but we cannot use it off-label without telling. So please give the authorization. It will be fully authorized and reimbursed on exceptional circumstances, if I could say so." But this is coming from the learning societies normally. So thanks a lot, Valerie. It was clear and very interesting because we are sitting on a gold mine. So this data could be used. And here once more, the collaboration with the learning societies, with the patients associations, and so on, is very important because we are not, us regulators, in the right situation to organize the collection of the data to the best profit of the research, but other people have to help us. But the data is there. You just have to pick it. So this is the end of this session.
So good afternoon and welcome back, everyone. We start now with the second session, which is focusing on how to establish a framework for ultra-rare sarcoma. And we will start with Teresa De Rias, Scientific Coordinator of the Accelerate program, who will tell us about the experience from this program for the pediatric community. So thank you, Teresa.
Thank you so much. Hello, everyone, and thank you for the organizers to give me the opportunity to present our experience with the pediatric strategy forums that we organize at Accelerate. I do not have any conflicts of interest to declare. However, I want to disclaim that I am a believer in these forums. I wasn't at the beginning when I joined Accelerate, but after participating in several of them, and also being behind the scenes, organizing them and so on, I have come to see how they work and how useful they can be. I hope, however, that I won't sound as if I'm trying to spread the gospel. Accelerate, for those of you that don't know it, is an international non-for-profit multi-stakeholder organization that promotes drug development for childhood cancer. So what are these pediatric strategy forums and how could they apply to ultra-rare sarcomas? These meetings are multi-stakeholder meetings with an open dialogue that happen in a pre-competitive setting on a malignancy or on a class of compounds. And the idea is that we have there experts from academia that are specialized in the particular disease that is being the focus of the forum, or in the class of compounds that has been discussed. But we do also have industry representatives there from the companies that are developing these compounds or are interested in doing so. We have, of course, the regulators on the table as well, with the FDA and the EMA not only being part of the meetings, but being even co-organizing these meetings. And we have patient advocates, usually both from that have a general interest in clinical research in pediatric oncology, but also that have a particular interest in the disease that is being discussed, for example. And the idea is that all of these stakeholders are treated as equal partners in these meetings with the motto of no blame and no shame, and creating or trying to create an atmosphere of mutual trust so that we can really discuss all together. As I said, the involvement of the EMA and FDA is crucial for the success of these meetings, and we have always independent funding, so we don't receive any funding from the industry to help keep the process transparent and feasible. Why are pediatric strategy forums needed? I don't need to convince you that the setting of the personalized medicine era has brought a lot of opportunities, but as you have also been discussing earlier this morning, it comes with a lot of challenges for drug development. And if we add this to the difficult setting of rare diseases, as is childhood cancer, then the result is that we do have a lot of problems. Many of the challenges were also mentioned already earlier this morning. So the forums are a tool to tackle some of these problems, and the objectives are to share information between all the stakeholders. In other words, trying to break down the silos and really sit everyone together on the table, to evaluate the available science, and to inform pediatric drug development strategies and subsequent decisions. So the aim is to improve the selection and the prioritization of innovative drugs that are evaluated for children and adolescents with cancer. And this, of course, needs to be driven by science and tries to meet the patient unmet needs. How does this translate into the practicalities into real life, let's say? Well, first, the topics are decided by the multi-stakeholder pediatric oncology community. There is a proposal period at the end of the year for several weeks. People can just propose what they think are hot or needed topics. And then there is a voting at the Accelerate annual conference to decide which concretely will be the next topics of the forum. We have an oversight committee that is responsible for overseeing all the forums. And we also have a dedicated program committee for each of the forums where we invite the most relevant experts of that particular field. The forums take place in 2 days, and now after the pandemic, we have transitioned to a hybrid model. These are closed meetings. We invite people to attend, the experts that they want to be there, but there is also the possibility to attend by expressing interest. And usually we have between 150 and 200 participants. About 80 of them are in person. I write preparation, but because of course the success of these meetings come thanks to a lot of months of preparation with many meetings with the experts, trying to define focused questions and concrete expected outcomes for the forum. Otherwise, the meetings result in too much talk, but no real concrete action derived from them. And as I mentioned before, the funding is independent, so it comes from charities, from NGOs, never from the industry. And also we have in-kind support by the EMA and by other institutions. We always publish a summary online right after the forum, and then we also publish the results, the main conclusions of the forum, usually in European Journal of Cancer within the next 6 months of the forum. So with all of this sounds all very good and nice. And if you're an optimist, maybe your expectations of how a forum looks like, it's like this, like happy children playing, working together. If you're rather a pessimist, maybe you will expect something like this, people fighting because they are very different and have different interests in mind. And probably the reality is something in between. It's like a UN convention with people with very different interests, and perhaps that don't necessarily like each other, but sitting together in the same room and trying to achieve common goals, in this case, benefiting children and adolescents with cancer. In spite of this, of course, there is always a huge elephant in the room because we have so many different stakeholders. There is a clash of interests. There is no way around it. We have, of course, the egos of the people, especially of us academics, with perhaps a particular research interest, and that can stand in the way. Of course, there is the bias of the financial interests, especially from the industry. Even the regulators can have their own constraints and limitations, but they are an essential part of the discussions. And sometimes it can be different. It can be difficult to think of the bigger picture, but it is what these forums are about. It's trying to strategize and prioritize what is best for our patients. And in spite of all of this and acknowledging all of this, somehow it works and the elephant can be tamed and it becomes a baby elephant. And the fighting that does happen is actually rather cute and I wouldn't say harmless, but it's not so horrible. So collaboration for the win. I think these meetings, the most important thing is to know that they do work and they are feasible. We've organized already 12 of them over the years. The first one started in 2017. And as you see, and as I've mentioned before, there are basically two types. Those that focus on a concrete class of compounds like ALK inhibitors, checkpoint inhibitors, or BET inhibitors. And then there are those that focus on a concrete disease, for example, on acute myeloid leukemia or on midline diffuse gliomas. This is one that we will have later this year. And at the beginning, they were all happening in the EU. But now we have started to alternate with North America. So actually the very next forum that we'll have will be in Canada. Overall, over 130 assets have been discussed over the forums and more than 1,000 participants have attended these forums, which for pediatric oncology is quite remarkable. These are some of the publications that have derived from the forums. All of these are in European Journal of Cancer and are open access, so you can have a look if you want. And to do a bit of further bragging about this, again, we are endorsed and encouraged by FDA and EMA and are cited in their guidelines. This is all very nice and good, but we have tried to measure the concrete impact of the forums, so that it's not that we are only convinced that they are good, but to see that they are actually producing some good for the community. And we've recently published these two papers in the Lancet Oncology and in GNCI, a bit trying to measure the perhaps unmeasurable. It's difficult, as you know, to measure the real impact of this policy meetings, or it can be difficult, but we've measured a number of variables, of course, number of participants and products, but also the pediatric investigation plans that were established before the forum and after the forum, same with the waivers and the clinical trials classified by relevance by the experts in the field. And we've also asked about the perceptions of industry participants, specifically about the impact that the forum had on their company decisions, and also on the perceptions of academics and how the forum impacted on the decision, on the prioritization of drugs in their academic trials. And we can conclude with all of these that the forums again do work and do have a positive impact on the pediatric oncology drug development. I will give you, to finalize, a concrete example of a forum so you can see, you can have a better idea. And this is the one that we did on mature B-cell malignancies back in 2017. So there have passed several years so that you can see understand the impact better over time. This forum was conducted out of the unmet needs of patients, as all of them. There were two particularly relevant unmet needs, or still are. One is to develop innovative treatments for incurable patients. This disease has a very high proportion of cured patients. However, those that relapse do have a very bad or can have a very bad prognosis. And the other is to reduce the high acute toxicity of the current frontline therapy, which again has high cure rates, but is extremely toxic. In the forum, some of the main conclusions were that there is a need to de-escalate at low risk in frontline therapy, but this can only be undertaken with an effective salvage regimen, which doesn't really exist. And there is a priority for developing treatments for relapse. The issue is that there is a very small number of patients, which of course is something fortunate that there aren't so many patients that relapse, but for clinical trials terms, this is very challenging. So a global strategy is needed. It's not enough to work on a national or even a European level. And also the combination approach was suggested rather than the monotherapy. There was an actual consensus between the clinicians, which can be very difficult to reach, but it was reached on which drugs to prioritize, which were the antibody drug conjugates, CAR T cells, and T cell engagers. And out of this forum, out of the discussion, there was the consensus reached that we needed a global academic trial, which has now concreted after these years in the Globe NHL and is about to start recruiting patients. This is an international, or actually intercontinental academic platform trial for relapsed and refractory BNHL. And as you see, the COG from the US, the C17 from Canada, ITCC in Europe, and ANSCOG in Australia participate to the trial. These are the main research networks from the different continents. And this trial is going to study the drugs that were actually prioritized by the forum. Another important conclusion of the forum was that there was a need to prioritize the drugs. There were too many pediatric investigation plans, so it was an objective that this number was reduced and the most important one pushed forward. And therefore having medically and scientifically justified waivers. And this is one of the things that we measured. And indeed, you can see how before the forum and after, there was a huge decrease in the number of PIPs, of pediatric investigation plans, and an increase in the waivers. So to summarize, in my view, and again, I'm a converted believer, these forums are not only about blah, blah, but they actually come with action and concrete results for patients, which is our ultimate goal. In a nutshell, the pediatric strategy forums are multi-stakeholder meetings on a specific malignancy or on a class of compounds. There is an atmosphere of no blame, no shame with mutual trust. Otherwise, these meetings would be impossible. We try to prioritize innovative drugs for children and adolescents with cancer, always driven by science and trying to fulfill patients' unmet needs. And they do work. We've conducted 12 so far with concrete actions and outputs. And we think that they can be a model for other rare disease settings beyond pediatrics, which can make them attractive or interesting for this setting of ultra-rare sarcomas. And this is all on my side. Thank you so much.
Thank you very much, Teresa, for the presentation. I think that for sure this is a model that can be considered also in for other rare tumors. And yeah, we leave time for questions at the in the last session. Now in the interest of time, we move forward. And I'll introduce Anna Maria Frezza, medical oncologist from the National Cancer Institute of Milan, and Professor Judith Beauvais, professor of pathology here in Amsterdam. They will tell us about how to collect retrospective data to be used for developing new treatment in ultra-rare sarcomas. Thank you.
Thank you, Silvia, and thank you, EMA, for hosting us today. Okay. So those were our disclosures. Very quick. So if you stick to the definition that Silvia showed earlier on today of ultra-rare sarcoma, you end up in having approximately 80 different types of ultra-rare soft tissue and bone sarcoma on which you need to take clinical decision every day. If you look on PubMed to retrieve evidence to support this clinical practice, and in particular, if you look for already published prospective studies of any kind on singular histologies assessing the activity of medical therapies in this disease, you will find 24 prospective studies ever run, mostly represented by phase 2 studies, which unfortunately inform us on only 9 histotypes and which were not enough to result in drug approval in the EU. At the same time, you will also find 23 retrospective studies on single histologies deriving from large collaborative networks of worldwide sarcoma reference centers, enrolling a reasonable number of patients, more than 50 in all cases, and providing us information today on 21 ultra-rare soft tissue and bone sarcomas. So in this slide, you only get a few examples of those large collaborative studies that we have prompted over time in order to explore the activity of currently available medical therapies in single ultra-rare sarcomas. These studies and these results are often for us as clinicians the best evidence you can get in this disease, and are actually the data that we use to inform our patients every day and take with them decisions on their treatments. So overall, why retrospective observational studies are so relevant for our sarcoma community, and I would say more in general for ultra-rare cancers? Because they often represent the only way to build evidence on the activity of available medical therapies in a reasonable time frame. This is, of course, essential to inform our practice, but it's also very useful to generate hypotheses that can prompt new clinical studies. They can serve as external control for prospective studies, especially in those circumstances in which randomization seems to be unlikely to happen. And we hope they could also provide a kind of benchmark on the available medical therapies, which can be somehow useful to approve new potentially active compounds. Although we know how relevant this tool is in ultra-rare sarcomas, we also know how many limitations the retrospective nature of this study implies. And because of this awareness, back in 2021, we established an international multidisciplinary group which has been working to improve the methodology of retrospective data collection and to establish the minimum criteria for an high-quality retrospective observational studies on the activity of medical therapies in ultra-rare sarcomas. This consensus has been joined by clinicians, biostatisticians, epidemiologists, and of course, basic researchers, and is also been shared with patient community and translated in a consensus which was published back in 2022 and represent for us today a guideline that our community follow whenever we want to put in place this kind of effort. During the discussion, we touched 7 different items which we felt were the most relevant when it comes to retrospective data collection, starting probably from the most challenging one, which is ensuring the quality of pathological diagnosis of all included cases.
Okay, yeah, thank you. My name is Judith Beauvais. I'm a pathologist in Leiden, not in Amsterdam, but in Leiden. I think it's my mission today, since I'm the only pathologist in the room, to try to convince you that it is very important to involve pathologists when designing clinical trials, but also for retrospective data collection. And why is that? That's because sarcomas are, as we all know, rare. There are many different subtypes. If you also include the benign subtypes, then it's more than 150, and they're all very much looking alike microscopically. 20% of them are ultra rare, and this includes 78 ultra rare sarcoma subtypes. So it's quite a challenge for pathologists that do not regularly see these kind of tumors to make the distinction. Concordance rates after pathology revision are quite low, 56 to 73%. And the major discrepancy rate is about 10%. So that's a discrepancy between the diagnosis made in an outside center and the diagnosis made in a reference center. So that's quite high, which illustrates that the diagnosis of sarcoma for pathologists is quite difficult. So how do we ensure the quality of pathological diagnosis in retrospective study? Because it is, as we said, the starting point. That's why we think that the pathology diagnosis should be confirmed by an expert sarcoma pathologist in the sarcoma reference center, because there pathologists have enough exposure and to gain the expertise to adequately diagnose these lesions. Up front, before we start the data collection, there should be consensus by a group of pathologists about the essential diagnostic criteria for the ultra-rare subtype that will be studied. These diagnostic criteria should be based on the WHO classification, and they should be dealing with the morphology, the immunohistochemistry, and the molecular alterations, because it's also always a combination of these three that sets the correct diagnosis. Sometimes there are very difficult cases that do not adequately fit, and for those cases in the paper there is a central pathology review proposed, and with the advent of digital pathology and most labs transferring to digital pathology, it should be much more easy than it was in the past. And also, when we do a retrospective data collection and there are questionable or uncertain cases, they should be excluded in order to have the series as homogeneous as possible. And this has led to the recommendations as it is in the paper.
So beside the quality of pathological diagnosis, we also discuss which centers should be contributing data to this kind of effort, and we all agree that only sarcoma reference center defined accordingly to the criteria, which are also recognized by the European Reference Network, should provide this kind of data together with sarcoma centers which belong to official national network, and this is essentially to ensure that the correct degree of expertise on the disease. We have a very long discussion about the assessment, the radiological assessment of disease response to the treatment, but also the radiological assessment of disease progression prior to treatment start, which is very important, especially when it comes to sarcoma with an indolent behavior. And we all agree that this must never be based on report or clinical chart, but always on imaging review performed by a dedicated sarcoma pathologist. We then identified some items that cannot be missed in the CRF, or at least explored with dedicated survey, such as the schedule of patient follow-up across the different institutions to ensure the consistency across contributing centers. And also, for instance, the name of the institution in which the patient received the treatment. And this is very useful to avoid data duplication. We selected overall response rate, PFS and OS as the best endpoint to be used in these retrospective studies. And of course, we all agree that as a general matter, but even more for ultra-rare sarcomas or ultra-rare cancers, every type of result should be published, including negative one, as they can be very informative. So in 2023, we put in place for the first time this recommendation, and we translated them in our first practical attempt. We conducted the first study, a retrospective study on an exceedingly rare sarcoma subtype. Thanks to a retrospective data review in a time span of 20 years, we put together 400 cases of patients with localized and advanced disease treated with available medical therapies, which was very useful for our community to inform us on the natural history of the disease and also about the activity of available medical therapies.
This just illustrates what I just explained. So we have three features that we look at when we make the diagnosis. So the morphology is described and is quite classic for these subtypes of the study. This subtype has a specific translocation which leads to overexpression of MUC4. So the immunohistochemical positivity for MUC4 is essential to make the diagnosis. So that's also a criteria that we ask for inclusion. And if MUC4 is not available because it's not available in the lab or it was not done in the past or for any other reason, then a molecular analysis and the demonstration of the fusion is also available. Just as an example on how we define as a pathologist, as a group of pathologists, the criteria that the diagnosis should be adhered to.
This study was joined by 28 sarcoma reference centers worldwide in the EU, US, Australia, and Asia. The data collection was performed through an electronic CRF built on REDCap, which is now available and fully reusable for upcoming studies. And the centralized quality check was used to avoid data duplication, to assess consistency in the procedure across contributing center, and also to identify possible other sources of mistakes. And the results of these studies will be published in the next few weeks. We are already planning a new effort of this kind, again, to provide further information on another ultra-rare soft tissue sarcoma, the adult type rhabdomyosarcoma, which is poorly covered by the currently available literature. And this study, which is scheduled for 2024, will be run in an even more structured framework, which is the push platform mentioned by Silvia this morning, and which will be presented by Lorenzo D'Ambrosio later on today. So in conclusion, the aim of this presentation was to summarize for you the work that we have done to improve as much as we can the methodology for data collection in observational retrospective data studies for ultra-rare sarcomas, which again represent for us as clinicians the base of our everyday clinical activity. So it will be our aspiration as community, and for community I mean clinicians, statisticians, researchers, and of course patients and patient representatives, to be able to use these same data, at least to have them considered as external control for prospective studies, at least when randomization is very, very difficult. So our question for today would be, is there anything else we can prove to further improve the process and to finally reach our goal? Thanks again for listening.
Thank you very much to Anna Maria and Judith. Of course, this could be the content of another workshop, but it's very important for us to understand if what we are doing is good enough and in which way we can improve it. So now we will have...
Can I ask one question?
Sorry.
So to Kit Roos, so if you saw the presentation and you see the effort that they're making to make retrospective data basis from this ultra-rare cancer, would you consider this a valid control arm? I mean, you explained that you can have data sets even retrospectively. Now that we are here, I'm just wondering what you think.
Maybe not a direct answer. I've actually looked at the presentation, but I've also read the publication in preparation of today's meeting. So I think it's a very good effort to have high quality data there. The question on does it serve as a control group will depend on the other side of the coin. What research question is to be answered, and will it fit? So, but my first concern would probably not be the quality where that's now being achieved. I think those are for me, they're always two separate questions. Needs to be fit for purpose.
And if I may, we will touch upon that in the panel discussion. From a regulatory point of view, we try to compare with an endpoint that is really independent as possible from the prognostic factors, so to the biases that could occur whichever we try to control in terms of prognosis. So PFS and overall survival are interesting endpoints, really supportive, really corresponding to a benefit, but you never know whether the two groups are fully comparable, even for characteristics you cannot control for. So overall response rate is probably the most robust and primary step to compare, because you do not observe responses spontaneously. Then, if you observe more responses, I would say that we go for other criteria, but the cardinal endpoint on which every effort should be made to make it as robust as possible is the response rate. And the time of response, the duration of response are a bit more sensitive to prognosis, but are interesting as well. And unfortunately, the more we approach the true clinical benefit, the less we can trust the comparison, because the more sensitive it is to prognosis. But this is a whole package. But I would say that the state of our guidelines for the time being is to say that response rate is probably the first on the top of the list of the endpoints to be investigated.
Okay, thank you. We will go on later during the panel discussion. So now we have Pan Patsiarka from Anti-Cancer Fund that together with Sandrine Moreau from the EORTC will tell us about the experience on repurposing pilot, in particular the example of sirolimus in epithelioid endothelioma.
We have no conflict of interest to disclose. I have difficulty to get it. Here, more than a year ago, the working group led by Sylvia considered of repurposing sirolimus for EHE patients based on the fact that there is unmet need definitely for these patients, the activity observed by clinicians when using sirolimus in these patients, and also based on preclinical and clinical data that were available. As pointed already out, this EHE disease is ultra-rare disease, and currently for the patients, most of the patients, 50% of the patients presents already at the time of the diagnosis with a metastatic disease, and for unfortunately those that have a paraneoplastic syndrome, the life expectancy is less than a year. There is no currently specifically drug approved for the EHE patients, but what clinicians could use is eventually the drug that has been approved for other soft tissue sarcoma, but there is a very limited activity for these patients, or going for an off-label use, but this is strongly dependent on the national regulation. We saw this morning in France that there is something planned for the off-label use, but it's not always the case in all EU countries. In terms of sirolimus have shown activity, this is an mTOR inhibitor and is currently used for preventing already rejections in patients with renal transplants. This is also a drug that is recommended to be used in advanced HAE by the ESMO guidelines, and has been used in the off-label already by some clinicians when possible in their country. Some preclinical data that have been conducted in a model, in patient-derived model, show comparing doxorubicin versus sirolimus, that there was a negligible activity for doxorubicin, but sirolimus induce at least 80% of volume inhibitions in the tumors. And there have been also clinical data based on retrospective series of in Italy on 38 patients, of which 13 patients had already paraneoplastic syndrome, and showing that there was a partial regression or stabilization in at least 86% of the patient. For all these reasons, we decided to go for an application and getting a scientific advice from the EMA on the repurposing of sirolimus. And we had a meeting that took place in June 2023.
So for the repurposing pilot, which emerged from a previous work done by the STAMP group, we assembled effectively a coalition of clinicians, patients, patient advocates, not-for-profit organization, the Anti-Cancer Fund. We marshaled the retrospective data, the preclinical data, to create a dossier. The only thing that we could not do was convince the marketing authorization holder of the of sirolimus to join us. In Europe, there is only one marketing authorization holder for the drug. In the United States, there are several. But regardless of their involvement, we went for the scientific advice process. We had a pre-meeting and then a full scientific advice meeting. And obviously, the key question that arose in the meeting is about the lack of prospective data. We've made plain how difficult it is to create randomized prospective data in a rare population like this. And at the end of this process, I mean, it's been quite positive engagement with the regulator. We are faced with essentially two pathways to explore. One of them is to explore the exceptional circumstances route, which can lead to a new marketing authorization. But for that, you need either for us as a collaboration to become a marketing authorization holder, which we're none of us in a position to do, or we have to convince the marketing authorization holder to join us. And we have tried, but they're not interested. The other pathway is the pathway that leads to a label extension, so not a new marketing authorization holder, but a type 2 variation of the existing marketing authorization. And to do that, we obviously need to discuss the prospective data collection, and we hope to do that in a subsequent scientific advice meeting. But at the end of that process, we are faced yet again with the fact that only a marketing authorization holder can apply for a label extension. So whichever pathway we take, we are faced with the fact that we don't have a company that's interested in working with us on this. So this is a huge constraint, and this is not unique to sirolimus and EHC. This is a problem that's faced for any generic product seeking a label extension. Now, we are aware that the regulations are up for grabs. There's new regulations being proposed, and one of the articles in the new regulations is Article 48, and that, in theory, will enable the regulator to seek a label extension without the marketing authorization holder initiating the application. So for us, this is a huge issue, and actually our experience has underlined why Article 48 is so important going forward. Now, in terms of the data generation process, we have to go to scientific advice. We can look for registry data collection. There's already a registry collection going on in the UK and in Italy, and we will seek scientific advice on that. We've already had some discussions about how many patients, what kind of endpoints, and so on. It could be that if we develop the push platform, which has been mentioned already and which Lorenzo will discuss this afternoon, we will be able to come up with an innovative method of collecting external controls that we can compare to a group of patients in a study. Or there may be some other approach that emerges in the future. But at the end of all of this data generation process, we either have Article 48, in which case there's a prospect of a label extension, or we again hit the obstacle. So this is our key thing, that we have to find a way past this obstacle. So, yeah, I think for us that's a key issue. Our questions then for the panel later on. Obviously, we want to formalize the requirements for prospective data collection. And in terms of Article 48, we need to know, is this a realistic proposition in a time frame that's meaningful to the patients we have at the moment? Thank you.
Thank you very much, Sandrine and Pan. Again, good question for the panel discussion. Now we move forward and we will have our colleagues, medical oncologists with experience in sarcoma from Dana-Farber and Di Wagner, who will tell us about prospective studies in ultra-rare sarcoma with the example of nab-sirolimus in PEComa. Thank you, Andy.
Thank you again, Sylvia. It's a real pleasure to be part of this discussion today. And I'm going to present an example of using a similar agent that was successful, but with many of the same challenges. But I want to highlight some of the unique features of this particular disease and the study that that may have led to a different result. These are my disclosures. So the disease I'm going to reference is called PEComa, which stands for perivascular epithelioid cell tumor. This actually is a family of different tumors, which include among it some non-malignant conditions such as angiomyolipoma or lymphangioliomyomatosis. It also includes an entity called PEComa, which can be benign or malignant, and other similar tumors. These tumors were defined pathologically in the 1990s, and they have very similar morphologic features and also immunoactivity for both melanocytic and myogenic markers. Now, AML and LAM can occur sporadically in patients, or they also can be associated with the inherited syndrome tuberous sclerosis, which is characterized by inherited loss of the tumor suppressors TSC1 or TSC2, which normally act as negative regulators of the mTOR pathway. And just not to go into the biology too much, but TSC1 and 2 suppress the mTOR pathway. And when you have loss of those tumor suppressors, you get hyperactivation of mTOR, leading to activation of downstream pathways that drive cellular proliferation and survival. But rapamycin and its analogs can inhibit mTOR and turn this pathway back off. Now, malignant PEComa is an ultra-rare sarcoma with an incidence of less than 1 per million people per year. In the United States, we anticipate there's about 100 new cases a year. It has a striking predisposition for women compared to men. Primary site can really occur anywhere in the body. The most common sites that we see are either in the uterus or the kidney. But like with other sarcomas, it can metastasize widely. And historically and anecdotally, it has been thought to be poorly responsive to standard cytotoxic chemotherapy. So because, though, of the similarities, the morphologic similarities to LAM and AML, and because of their reported activation of the mTOR pathway, as well as a little bit of preclinical data suggesting that mTOR was activated in PEComas as well, we and others began the off-label use of mTOR inhibitors in patients who had advanced disease. And we described a small case report of 3 patients who had significant benefit with the use of sirolimus or everolimus, an example demonstrated here of a patient with baseline pictures on the left and on treatment pictures on the right, showing significant reduction in size of the tumors. At the time, the next generation sequencing was not available, and these are very big genes, the TSC1 and TSC2 genes, so it was very difficult to define whether these patients had underlying alterations. Additional case reports followed either at the same time or in subsequent years, including other reports of patients who've had benefit from mTOR inhibitors, either in the US or in several European countries as well. And I would say that really globally, mTOR inhibitors were off-label use was adopted as the standard approach to managing this disease, but there had been no drugs registered for this purpose. And we, and as well as colleagues in Europe, tried for many years to find a pharma partner who was interested in using one of their existing drugs or even investigational drugs that were being developed to study it prospectively and ran into the similar challenges that we just heard about with those companies. Fortunately, though, in the mid-2010s, I was in contact with someone who was forming a new company and had acquired the rights to nanoalbumin-bound sirolimus, so sirolimus bound to these small particles of albumin, who was looking for an indication to study. And so we designed this study in malignant pecoma using nab-sirolimus. And there were 31 patients who were evaluable in this course of the study. They could not have had any prior mTOR inhibitor. We enrolled the patients over the course of about 2.5 years. It took to enroll those 31 patients across 9 sites in the United States. And the primary endpoint was overall response rate with other key endpoints that included duration of response and PFS. And the response among those evaluable patients was a 39% confirmed response rate. You can see that in the waterfall plot above. The colors reflect retrospective genotyping that we performed of the tumors, which showed a strong association with TSC2 mutations, but also responses in tumors that had TSC1 mutations or no identified mutation. And the spider plot shows prolonged duration of response. The safety profile was very similar to what had previously been known with the oral mTOR inhibitors. And these data were used as part of the package that did lead to FDA approval in November of 2021. So this has become an approved tool. We still can use off-label in the US, off-label use of the other inhibitors, but this is one that is now indicated for the treatment of advanced malignant pecoma in the United States. So I just wanted to end with this idea that there's I came up with 3, there maybe are more legs to this stool, but without any one of these, the stool would topple. And so we had a disease in which there was a commonly altered pathway. If that was a rare alteration, we may not have been seen a response rate like we did. Paired with a drug that both could inhibit that altered pathway and was well tolerated by patients. We found a partner in industry that was willing to conduct a study in this ultra-rare disease. And I think importantly in the single-arm small study, there was activity that was objectively demonstrated by significant tumor shrinkage. But without any one of these 3 items, I think we would be left still in a very similar situation as we just heard about for EHE, which is some evidence from anecdotal treatment of patients that there's activity, but harder to get to a point where we can get to a drug that's licensed for this use. So the question I would pose, just highlighting one of those, is in the example I presented, the reduction in size of tumors was a marker for drug activity. But in these ultra-rare populations where randomization is not feasible, how do we demonstrate efficacy when tumors are controlled but don't have significant shrinkage? And I look forward to that discussion later. Thank you. That was it. Oh, you can gain control.
Thank you very much, Andy. And now we will ask Annalisa Trama, epidemiologist from the National Cancer Institute of Milan, to talk about prospective registry. We already mentioned prospective registry several times as an option to collect data to add to prospective studies. Thank you, Anna.
Thank you, Silvia, and good afternoon to everybody. I had no conflict of interest, and I managed to go directly into the slide fixing the problem of the mouse. So first test was passed. It's a pleasure for me to introduce you the registry that since the last 4 years we have been starting in the framework of the European Reference Network on Rare Adult Solid Cancers, EURACAN. You know, this includes 10 different families of rare cancers. We initiated with rare head and neck cancers, sarcoma and ultra-rare sarcomas, because these were some of the families in which needs were high, where initiatives were ongoing. And so we felt that the registry could really represent an attempt to harmonize and coordinate initiatives that were also ongoing at national or single institution level. So we really try to leverage and coordinate widespread activities at European level. This is the outline of my presentation. That is not by chance. I followed basically the major area of recommendation that in the different workshop that in the EMA were performed about registry were followed. I just added the objectives because I think that the objectives represent one of the main issues to start a registry in the proper way. What we are discussing here, it's a hospital-based registry, so it's a clinical registry that is based on expert centers. The objectives are quite broad because, of course, the registry is intended for a long-term data collection. So we need to be broad and we have to have a long-term vision. But I think that you can already see that these broad objectives often, correspond to what also the EMA is mentioning as some of the possible role that at least in a pre-authorization phase, these are information that a registry, can complement in terms of evidence. The registry is not collected sample. However, we are including in the registry database information about available samples and available imaging so that we can definitely have an infrastructure that can easily get an idea about feasibility of translational studies in case, in futures there could be the need to test, for example, in some sample, new biomarker. Of course, in order to provide a flexible infrastructure that will enable to accommodate updates, new information and new knowledge, we have envisioned a registry with certain type of data set, and I will go into it in a while, but it is intentionally done to be considered as an ad hoc registry, so that in addition to the common data set with which the registry starts in case of any specific new discovery, or if a specific study is needed, we can add on to the common data set what is needed for a specific study in order to be flexible enough and to already envision changes that, of course, in such a long-term infrastructure are needed. Data elements, there is a lot of discussion about which is the best data elements to start a registry. I decided to take the example of the sarcoma family because within this we are basically studying two different use cases, the common sarcomas and the ultra-rare sarcomas. It has already mentioned, we initiated a registry on the epithelioid hemangioendothelioma. Again, these are just use case and example. What's the difference? The difference is that for common sarcoma, the type of research questions are different. Because for common sarcomas, the knowledge is higher than for ultra-rare sarcoma. And that's why we define a core data set, The core data set, of course, has been defined accordingly to international standard. We have data dictionaries because, of course, behind all this, there is also the worries and we want to ensure interoperability of data. That's why we always work with international standard. And since for common sarcoma, there were already initiatives ongoing, we decided to leverage common data model in order to harmonize data set already available, but also because this will allow us, to rely on already available tools and to make the registry possibly, of interest also for other international collaboration that are using the same data model. For ultra-rare sarcomas, we really had to go into a comprehensive data set because the type of research question needed more detailed type of data, but still, methodologically speaking, we are still a respective standard. We are still providing, fair approach in order for everybody to understand what we are doing. And no data were available, so we had to develop a new electronic CRF. How we develop the registry elements? Of course, these were developed based on expert, but I have to admit that patients were really, seated next to us when we were developing not only the data set, the full registry. We develop an ad hoc, in Huracan, we have a group of HEPACs, which are the European Patient Advocacy Group. So they were really standing behind us in developing, in deciding from which rare cancers family we had to start from. And you'll see they really provided also a lot of input also for the governance. The second point has been touched earlier on, is about the registry quality assurance. Of course, there are standard indicators for quality. We are picking up those related to conformance, completeness, and plausibility. We are checking basically internal consistency, but also, consistency over time. We are searching for benchmark, internal and external benchmark. I don't want to go into the details, because what I think it's important for me to explain in this context is the peculiarity of this registry. This is a federated registry, so we don't have a centralized database. And this was due to two main reasons. The first one is the privacy, and especially, the National Competent Authority, that at least in Italy didn't allow at all to have a centralized registry. So we had to go into a decentralized, the federated approach, which basically means that the data are stored at the hospital level. Okay, so at the place where the data are collected. This turned out to be something really relevant, also because we know we are here, and we really want to share the data, but still there is a lot of skepticism around, the data sharing. There is a fear to lose control of data. So the idea of having a decentralized registry, with the registry that remain at the hospital, really increase, I would say, the trust in what we are doing. Why I have anticipated the registry infrastructure talking about quality? Because even if it is a decentralized registry, we clearly ensure quality checks not only at the local level, providing supports for performing data quality checks at the local level, at the entry level. We provide training, we provide guidelines, but we do have also a centralized way of controlling the data, and we still have a person dedicated that will develop reports, will discuss with the centers. So there will be a continuous and, I would say, stepwise approach to really always increase the capacity of those collecting the data to harmonize and having the best data available in the registry. The last important point is related to the governance. It has been said the registry can be used for different purposes and by different users, which I think it's important. We want this registry to be reused by everybody that might needs it, because it's such a huge effort to perform it that we really need it. That's why, again, we develop a governance together with all the different steering committee and board members of EURACAN, with patient advocates, and of course, with the expert of each domain, in order to clarify which are the rules for accessing the registry. And what I think it's important is that the governance and therefore the data accessibility is centrally managed. I initiate my speech clarifying that, we were really trying to harmonize and coordinate activities. So we really hope that a central managed data access process can facilitate all those interested in getting the data in really, trying to use the data. The data access rules clarify who can access, and again, since we want the registry to be used, you can see that we have already envisioned that also competent authority can request to access the data, but also pharma company. And we are envisioning also the idea that, we could get funding for data that are provided. We discussed this at length also with patients, because these registries are costly. So we need to find a way to sustain them. It's not for profit. I used to say that it is a low profit. We need funding to reinvest into, the integration for the management of the registry. The legal basis of the registry, we learned in the last 4 years that this is really country specific, although in most of the countries, the legal basis is the patient consent. So we develop patient consent, and especially, we always pay attention that the patient consent include the possibility of sharing the data with outside the hospitals and with different type of stakeholders and also with the international group, because again, we want data to be used and reused. used. And I will conclude with the example of the different way we are going to analyze the data, which are all approaches that are privacy preserving. The first one is the federated learning, that basically is going to use machine learning techniques that is basically training algorithm across decentralized database. The main principle of the federated learning is that, as I was saying, the registry is federated, so the database are at the hospital level. The federated learning implies that the algorithm goes to the data and only aggregated results comes out. So no data, only aggregated results come out. And that's why it is being considered as privacy preserving. Of course, it's new. There are pros and cons. And what we realized is that you need to have a core, a simple data set to analyze data with the federated learning, although you can do modeling, you can do a lot of the type of statistical analysis you are used to do with your statistical package. Of course, if the database is more complicated, like the one that we are developing for the EHE, again, what we are thinking, the database are decentralized. So in order to have a better capacity to analyze them, we will anonymize the data and centralize the data once they are anonymized. This will allow us to better understand this complex type of data set. But of course, we can also request only specific type of data set for specific research questions, so that every data source can be satisfied with the type of data that will be shared. And last but not least, I anticipated that we are also mapping into the common data, the HOMOP common data model, one of the registry, the one on sarcoma. This would allow us not only to use federated learning, but also simply to do federated query. And so we are trying in a certain sense to develop an infrastructure that could accommodate different needs, different research questions, different possibility to really trying to embark in a different project. And so, of course, I will conclude with my question that is really, do you think that we are in the right direction? Do you think that what we are doing is something that could be considered relevant and enough also, okay, for the idea of using this type of data? Of course, when this type of data are useful for specific research question for the regulatory assessment. Because I think that here, and also Lorenzo later on will explain, we are really trying to put forward some infrastructure that could be considered good or very good, as Kit was saying, also for regulatory assessment. And if you are wondering, we started the data collection for rare head and neck cancers in April 2022. We are already mapping the database for sarcoma in 4 different countries, and I'm happy to say that in December 1st, we launched the hemangioma registry. And I thank you for your attention.
Thank you very much, Annalisa, and all those who contributed to the registry. Of course, it's a lot of work, not only to maintain it and to include the data, but also to build the registry for each single disease. So it's really important for us to understand if this exercise is going to be helpful and what in case we need to include, to add, to change, to ameliorate. This said, I'll give now the floor to Lorenzo D'Ambrosio, medical oncologist from Turin, Italy, Gauthier Bush from Anti-Cancer Fund, and Rosalba Miceli, statisticians from the National Cancer Institute of Milan. They will present as the PUSH project, and yeah, I will not take more time, and I'll give to Lorenzo. Thank you.
Thank you, Silvia, and thanks for having us today. We will talk about these ideas that we are working on. These are our disclosures outside this presentation. And our aim was basically to maximize the knowledge from every single ultra-rare sarcoma patient with the aim to support the development of new treatments and improvement in outcomes and quality of life. Of course, you can say, okay, this is like asking for the peace in the world, okay? But as in John Lennon's song, you may say I'm a dreamer, but I think that I'm not the only one, since we are quite a lot today. So we start from these slides that I took from Silvia's presentation, because she elegantly highlights the main challenges of ultra-rare sarcomas in terms of not the possibility to randomize in a common manner, regulatory issues, few approved drugs, few interest of pharmaceutical companies. And we started thinking of a solution for this problem, among our research groups, among the EORTC soft tissue and bone sarcoma group, as well as among the ultra-rare sarcoma working group of the CETOS. And we easily saw that we are not alone in trying to find a solution, because this need is also on the side of patients. And we heard from you and Joss how this is important for them, from research funders, from biostatisticians, translational researchers, and as well as for regulatory agency. And we try to work on a solution for this project, for this problem, and we finally ended in this PUSH project that we are working on since a while, in the last months. And the prospective part of this PUSH project is called PUSH IT. So pushing ultra-rare sarcomas toward hope, an international trial. So our aim is to develop also some prospective trial in this field. And in the PUSH IT, we will try to take advantage of some of the features of a platform study, taking into account that we cannot use the same platform studies used in more common cancers like breast cancer, prostate cancer, where they have a common standard arm and several other arms where patients are randomized and so forth and so on. But we can take some of the features of these studies, like being faster in the development of new drugs, being more flexible, with the ability to add and cut arms to a main body of the study, in order not to start from the scratch anytime. And in the end, this might translate in being more attractive to new compounds, since we do believe that if all work in some way could also reduce the cost and increase the interest of new compounds in the field. So basically, our idea is to have a sort of master protocol as the main architecture of this project, a master protocol that basically will set some rules for a common framework, a common governance of the entire project, will contain, and we already put in the first version of this master protocol, some predefined design hypotheses for clinical studies. Of course, this design hypothesis cannot cover all the situation of ultra-rare sarcomas, but might represent a guidance for clinicians in the development of their idea. Of course, this might constitute a first ethical evaluation before moving on, and will contain some rules for data management and IT. Of course, from this master protocol, several sub-studies can stem. And all these sub-studies might be not only drug-specific, but since we are talking about ultra-rare sarcomas, isotype-specific, or even molecular subtype-specific, also following what Denis presented us earlier today. And of course, our main focus will be single-arm studies, since we are talking of ultra-rare diseases, but not only investigational studies, because you heard from Anna Maria's presentation how also being aware of the behavior of a disease, so knowing the natural history of the disease is so crucial, and also observational studies can be very important, and so forth and so on. But when talking about single-arm studies in sarcomas, we are facing a challenge that is the average low expected response rate that we discussed earlier on, is the best way to detect a drug activity in a single-arm study. So we can try to exploit as much as we can the data we have to better interpret the activity of a drug in a disease. And one of these ways could be the improvement, the exploitment of the use of external control. And in this way, we will try to add external control group to any study, considering to have the same inclusion criteria, the same time points for radiological assessments, the same items to be collected for the CRF for this patient enrolled in the control group, as well as in those one enrolled in the investigational group. We can count on more than 50 academic sarcoma sites around the world. This is just a graphical representation, it's not an actual representation of every single site, okay? But we can foresee several different situations. One might be we can have an experimental treatment tested in the US, but not available in Europe, by instance, and we can serve as external controls. Or we can have a more scattered situation with some site from the PUSH project involved in the experimental treatment and some other we enrolled as standard of care. And also a more jeopardized situation where you can have a center treating patient with an experimental treatment, one with standard of care, and another with just observations. This might be the case of less aggressive diseases. So several situations, several scenarios. And we have also to consider that the concept of external controls is expanding over time. So we are now more and more data coming from patients themselves, not only patient-reported outcome measures, but also data coming from patient registries reporting clinical outcomes. We have a growing amount of genomic and molecular data, and a few weeks ago, it was published also a paper on the rules to evaluate liquid biopsy responses in solid tumors. We are now all working on improving the ability to catch data from clinical charts. This is also due to the increased use of artificial intelligence to improve this process. And we, for sure, in the push it, we will try to take advantage as much as we can to already available data coming from clinical trials like academic trials, cooperative group trials, industry-sponsored trial, and also any study run within the push it will serve as a basis for future trials. And finally, we already mentioned today, there is a growing interest in real-world data that encompass also the prospective registration of off-label use of drugs like sirolimus in EHE. And Annalisa just told us how registries can be a powerful tool, and also all the effort done for the repurposing of drugs can be of help. So basically, anytime we will run an investigational trial, we will start a prospective observational trial in all the centers not involved in the experimental treatment. Of course, it's not realistic to have more than 50 sites open worldwide with a new drug. It will cost too much. And in that case, we will once again use the same rules for accrual of patients. And we will also consider the use of synthetic data. That is, once again, a powerful tool of growing importance that might help in improving the data and saving patient numbers. Okay. I just want to disclose that we are not formally against randomization. We are not formally against, randomized controlled trials. But of course, since we are talking about small numbers, we have to consider innovative measures, innovative way of running randomized controlled trials like unbalanced randomization, Bayesian randomization, discontinuation trials. And thanks to ALBA, we put already in this predefined design hypothesis also some options for very small randomized studies. And once again, we can be creative in the control group of randomized studies, like tweaks, like synthetic data, and so forth and so on. So in conclusion, this is of course a work in progress. The push it is aimed to exploit as much as possible the push platform potentials to maximize the knowledge from each patient, because ultra-rare sarcoma patients are out there. We still miss too many information from this patient, despite we all treat them. and we need to look for innovative approaches. And we do believe this might be also a starting point for other rare tumors, not only for sarcomas. And so in the end, my question to the field is whether we can aim to plan periodic meetings with EMA and even FDA to develop the Push It project, to develop the single sub-studies of the project in order to design these studies in the best way possible and increasing as much as we can the chances to finally end to a registration of a drug in case the study is positive. Thank you very much.
So thank you very much, Lorenzo. It's clear that Push It is a part of Push. Push is the full consortium in which we foresee the collection of retrospective data, translational data, and all kind of data we can. And Push It is the platform for the conduction of prospective clinical studies. For us, this is a very huge investment. We really want to be global, and we need to have a legal agreement that can help us being ready and fast, because one of the other problems we have not yet mentioned today is that often the new drugs arrive to sarcoma later, when the development phase is already in the middle or already done, and so they do not arise any interest. So we need to be ready on time, and we have decided all together, as Lorenzo said, that there are more than 50 institutions from US, Canada, Australia, Taiwan, Hong Kong, of course, Europe, that are really want to join. So we cannot do that alone. We need you. I mean, we need the regulatory agency, EMA, but also FDA, because as I said, we want to work together. And there are many aspects that need a constant consultation, comparison to be sure that what we are doing is good enough. And if there are no comments now, I think we can go for the break in time. We have 15 minutes, so we come back here at 3:13. So we can start 3:15, sorry. We can start on time, and we will have two presentations from FDA and NCI, and then we will have time for discussion.
So welcome back for the last set of presentations, and then we will go for the panel discussion. So we have two speakers for that last presentation session. So the first one is from the NCI, the chief of the NCI Center for Cancer Research in Pediatric Oncology.
I might go through some things more quickly, but what I was hoping to do is to update you on efforts that we have in the pediatric oncology branch, but more importantly also through NCI's Childhood Cancer Data Initiative, which was started in 2019. Okay, I don't think I'll have to go through rare cancer definitions, but really importantly, not only are they rare, but in the sum of it accounts actually for 25% of all cancers. And for many of these tumors, there is no standard of care and no clinical trials. Cristina Vivello from our group did a landscape analysis that showed, based on publications, as you can see in the right lower corner, there are many, many more publications in the past years than in the past, and there are many rare tumor programs based on an analysis of published literature and also an online search. But there is the need to bring these efforts together to get sufficient patient numbers and have standardized and structured data collection. In addition, especially with the focus in this workshop, as the CTAS defined ultra-rare tumors, and it was nicely published, an entity that rarity makes it extremely difficult to conduct well-powered studies. And when I was asked how many rare tumors are there, it is many. And if we want to make progress in all of these tumors and more quickly, I think we all have to work jointly. In 2017, with Cancer Moonshot funding, Drs. Riley, Abby Sandler, and myself established my pediatric and adult rare tumor network at the NCI. The centerpiece of it is a longitudinal natural history study that retrospectively, but more importantly also prospectively, collects molecular, clinical, and patient-reported outcome data on a natural history study. In addition, and I will mention it, we conduct multi-day clinics at the NIH that bring experts and advocates from many different places and patients so that they can get treatment opinions. The protocol is shown here. We get outside records, evaluate them, do extensive medical data extraction. Patients can enroll off-site or on-site, so they can come to the NIH Clinical Center, but also participate remotely. The patients do complete a number of patient-reported outcomes, including a family medical history and patient-reported outcomes. We do comprehensive molecular profiling, return the results to the patient, have a molecular tumor board, genetic counseling, and are creating a biospecimen repository for the tumors that we see, and obviously have the goal for interventional trials. As of September, I believe we had enrolled more than 570 participants, importantly with many, many different rare tumor histologies. We collected biospecimens from patients who undergo surgery at the NIH. We have worked on establishing PDX models, and we are establishing a genetic mouse model of SDH deficient GIST. We established partnerships with a number of advocacy organizations, and we have had in-person specialty clinics for GIST, chordoma, and neuroendocrine tumor. We are working on how to best analyze and integrate the data, and all of the specimens actually, and the data has been submitted to dbGaP so that it's available to the research community and anybody. What I want to highlight here is the different diagnoses that we encountered. We had what I would call 4, maybe 5 meaningful cohorts where we have more than 20 patients enrolled. GIST has been an interest of us for many years, neuroendocrine adrenocortical carcinoma and medullary thyroid carcinoma. Chordoma was really a new effort that we established in collaboration with the Chordoma Foundation, and we were able to enroll more than 100 participants. What we really learned from this is that if we want to make progress in many tumors, we really have to have a national and international, ideally, collaboration. Before I go to that international collaboration, I briefly want to highlight the approach of rare tumor clinics. We bring about 10 to 15 patients at government cost to the NIH Clinical Center. We bring disease experts, as you can see here in the picture on the right-hand side, surgeons, radiation oncologists, otolaryngologists. It depends on the type of tumor, advocates. Patients present, get detailed treatment recommendations, and I do believe this is a true benefit for the patients, but it also connects our research community and gives expertise that we don't have at the NIH. This shows, we started with the Chordoma Foundation with a workshop, developed a partnership, the clinics. We work with the Boston team, Shannon McDonald and Greg Cote, where there are regular imaging review and discussion of patients, which has been extremely beneficial to us. Dr. Riley was able to get a grant to start a SMARCB1 deficient chordoma, and Dr. Zvedekind is co-leading with Dr. Akshintala in the POB a phase 2 trial of 2 checkpoint inhibitors for SMARCB1 or SMARCA4 deficient tumors, which includes a cohort for poorly differentiated chordoma, but a number of other tumors that you see here. And importantly, this is an PEP-CTN, so a broad multi-site study that allows enrollment of both pediatric and adult patients. What I think we learned is, and I think that would be I think, critical for the ultra-rare sarcomas. One is that advocacy is critical, as is rare tumor expertise. I do think the NIH rare tumor clinics, where it makes sense, such as for a tumor like chordoma, can be highly beneficial and provide insight that would be difficult to get otherwise. But the partnerships are really needed to accelerate the pace to make progress, and multiple disease champions are needed if we want to not only have a registry, but also hopefully provide benefit to the patients. The NCI Childhood Cancer Data Initiative has established an effort with the hope to develop a national and international collaboration to help make progress more quickly. Sorry. So the CCDI was initiated in 2019 and announced at the State of the Union and provides funding for 10 years. I think we're now in the third or fourth year to focus on childhood cancer. And our NCI director at that time said, I want to establish a data community centered around pediatric cancers with the goal to expand it to adults, but to create a data ecosystem where we can aggregate and generate data, share data. And with that, Dr. Greg Riemann actually is the scientific director. He was recruited about a year ago, and that has made a wonderful difference for this effort. It is very active. And for today, I just wanted to mention two aspects that I think are critical to potentially the rare sarcomas. One is the CCDI Molecular Characterization Initiative, and then the National or International Rare Tumor Initiative. So one project for the CCDI was to establish a partnership with the Children's Oncology Group to provide state-of-the-art molecular characterization of tumors in children at time of diagnosis. To make this work, patients have to enroll on COG's screening protocol called Project Every Child. Results are returned to participants within 21 days. All of the analyses are done at Nationwide Children's. And within the first year, more than 1,000 participants were enrolled from 47 states, including international patients. And as of August 2023, more than 3,000 patients had been enrolled. This was started with CNS tumors, then soft tissue sarcomas, and most recently, rare tumors. These were all at the time of diagnosis, but now we are going to, which is obviously really important, also look at relapse samples, which could be a very unique resource. And with that, also clinical data are collected as part of the COG. And I do think this effort could help us potentially identify specific rare tumor patients of interest for an international effort, and also molecularly defined rare tumors for analysis, such as, for example, poorly differentiated chordoma patients. We are in the process of developing and would really look forward to collaboration with the EU and, of course, guidance from EMA and FDA, a national observational protocol for very rare pediatric and AYA solid cancers with the goal to expand it to hematologic malignancies. This will be longitudinally and prospectively evaluating the disease course of participants. We want to use the same, similar to the earlier presentation, a core data set so that we can compare different rare tumors and have expanded data sets as we develop potentially external controls for clinical trials. And we, in addition to our standard molecular clinical characterization, are hoping to do research molecular characterization. And we would be very interested in the feasibility of national and international molecular and clinical tumor boards, which I think could be, again, providing benefit to patients where there's very limited expertise in many of these cancers. So the patients currently would be enrolled through COG and Project Every Child, identified, and then we would enroll them. through self-referral. So this could be a patient from anywhere in the world. There would be two components: the molecular characterization, where there is clinical sequencing that is returned to the care provider and to the patient, research sequencing, and then clinical data collection, very similarly to what was described earlier, where there would be a core and comprehensive data analysis for specific subsets, and all of this data would go into the CCDI data ecosystem. I think the hope would be that, similar to what you described, while the coordination would be centrally and the resource for data extraction, but that there would be disease champions from anywhere in the world that could actually champion their own disease and potentially use this data infrastructure. We have started, and this is still in the beginning, but we had a wonderful meeting actually yesterday with the EU and several members of the expert group, and Dr. Ladenstein, who leads the EU effort on potentially developing a focused international research collaboration. And I was thrilled to hear there, and also today, that there are already core data sets that you have established. So it would be wonderful to see what these are, and can we really collect identical or near identical data so that we could potentially share this data and increase the number of patients with specific rare diseases. Of course, a collaboration with international collaboration would substantially increase the number of patients with rare tumors. It would potentially, we could use CCDI resources to help integrate and join the data. And I do think the international collaboration would also hopefully allow representation and more disease champions for rare tumor boards. And as it was mentioned earlier, the ultimate goal obviously is to get this to interventional clinical trials. And I want to highlight two examples that all of you know, obviously, but that give me a lot of hope that this is actually feasible. On the right-hand side, the wonderful example of the development of larotrectinib for NTRK fusion cancers, where it was a non-randomized study, pediatric and adult patients, and the approval was on the objective response rate that was, and the deep depth of response and the duration of response, with 12 pediatric and 43 adult patients. And then the more recent example of atezolizumab for alveolar soft part sarcoma through a multi-site study. And again, it was only 50 patients, only 2 pediatric patients, and it was based on the depth of the response, duration of response. And it was an approval that, even though there were only 2 pediatric patients, includes pediatric patients. So in conclusion, I'm so excited to see the efforts that are ongoing and already in collaboration with Europe and the United States. I think the regulatory efforts are very exciting, and we'll hear about this, I think, in the next talk. The CCDI is hoping to create a foundational infrastructure that can be utilized by through multiple sources, and we hope that this effort will contribute to more rapid progress in rare cancers, including obviously rare sarcomas. I think we all know that we have to work together to make progress, and this is from a book on rare tumors that was edited by Dr. Riemann and Dr. Smith. And I want to thank many involved. I keep this brief here, but it is many individuals within the NCI, outside of the NCI, but in particular, I want to highlight the collaborators and the advocates and the families who participate in observational studies when they don't necessarily gain benefit from doing so. Thank you very much for the opportunity to participate today.
Thanks a lot, Bridget, for this impressive presentation. It is important to make friends. As a matter of fact, we are here a bunch of friends, so it is a starting point, which is not bad. Thanks a lot. This presentation is exactly the type of initiative we should join or develop if they would not exist. But really impressive. Thanks a lot. So let's go to the next presentation, which is by Caitlin Tidings, who is a clinical reviewer for the sarcoma team at the FDA. Thank you, Caitlin.
Good afternoon, everyone. So as mentioned, my name is Caitlin Tidings. I'm a pediatric oncologist and clinical reviewer at FDA. I'm appreciative of the opportunity to speak today in hopes for continued future collaboration. So today I'll be providing an overview of the FDA's perspectives on rare cancer drug development, with an emphasis on FDA programs available to help mitigate the challenges we face in this space. Next slide, please. I have no relevant conflicts of interest. Next slide, please. So first, I'd like to give you an overview of what I will cover today. I will briefly provide a background on the structure of our division within the FDA, rare cancer definitions, as well as the current state of recent approvals. And I will focus most of my time on the challenges that exist in drug development for rare cancers and the programs designed to help overcome some of those challenges. Next slide, please. I thought it may be helpful to provide an overview of the structure of the FDA. You will often hear me refer to the OCE, which is the Oncology Center of Excellence. The OCE was authorized by the 21st Century Cures Act of 2016 and was established in January of 2017. The OCE unites experts across the FDA to conduct expedited reviews of medical products for oncologic diseases. So please bear with me, there are quite a few acronyms coming up, but within the OCE lies the Center for Devices in Radiologic Health, or CDRH, the Center for Biologics Evaluation and Research, or CBER, and the Center for Drug Evaluation and Research, or CDER. So CDRH helps assure that patients and providers have access to safe and effective medical devices and safe radiation-emitting products. We collaborate with its Office of In Vitro Diagnostics and Radiological Health for companion and complementary diagnostics. Within CBER lies the Office of Therapeutic Products, which regulates biological products for human use, including CAR T cells and other cellular therapies, gene therapy, and therapeutic vaccines used to treat cancer. And finally, CDER houses the Office of Oncologic Disease, or OOD, and they oversee the development of drugs and biological therapeutics, including small molecules, monoclonal antibodies, antibody-drug conjugates, all used to treat cancer. Next slide, please. This is a snapshot of the organization of the disease-focused divisions that comprise OOD. There are 3 divisions dedicated to solid tumors, 2 to hematologic malignancies, and 1 to toxicology and non-clinical review. I'm a reviewer within the Division of Oncology 3 and specifically on the sarcoma team. Notably, across OOD, there are over 20 pediatric oncologists and over 60 medical oncologists who work here. Next slide, please. So now that we have a general understanding of where the sarcoma team lives at the FDA, we can shift gears to discuss more rare cancer drug development. So let's start with how rare cancer is defined. And while there is no universally accepted definition for what is considered a rare cancer, the FDA definition of rare, defined by the Orphan Drug Act, is a disease that affects less than 200,000 people in the United States. It is important to note that even the most common sarcomas are considered to be rare cancers. Next slide, please. So over the last decade or so, 11 drugs or biologics have been approved specifically in the sarcoma space, as you can see summarized on this slide. I would be remiss if I did not mention tissue-agnostic approvals like larotrectinib that Dr. Wiedemann referred to. They're not included in this table, but they may also benefit sarcomas, and I'll be discussing this a bit later. As you can see, 5 of the sarcoma approvals were based upon improvement in time to event endpoints, such as PFS and OS in randomized controlled trials. However, several sarcoma approvals, including tissue-agnostic approvals like larotrectinib, were based on improvement in ORR in single-arm studies. While this table does represent some advances for sarcoma drug development, a lot more work needs to be done, which I think we all agree on and is why we're all here today. Next slide, please. As evidenced by the limited approval shown on the previous slide, there are several challenges in sarcoma drug development. Many of these challenges are common to all rare cancers. So this list provided is not exhaustive, but a few notable challenges are highlighted. First, as we all know, sarcomas are rare, and this rarity can impact not only our understanding of the disease, but the way in which drugs to treat sarcoma are studied. Small patient numbers, geographic dispersion, and limited or lack of timely access to molecular testing may cause obstacles to timely accrual. Sarcomas themselves can be very heterogeneous tumors, both genotypically and phenotypically, and due to the rarity of these subtypes, the natural history is often not well characterized, and the understanding of its pathophysiology and molecular characteristics are still being elucidated. Oftentimes, sarcomas that may be genotypically distinct are studied together in the same trial due to challenges accruing adequate numbers, though this may lead to difficulty in interpreting endpoints. Next slide, please. OCE has worked to combat some of these challenges through initiatives and programs collectively aimed to promote the development of safe and effective new drugs and biologics to treat patients with rare cancers. And while there are more, I will highlight a handful of programs that benefit rare cancer drug development. Next slide, please. At the helm of these initiatives is the Rare Cancers Program. In order to achieve the goal of promoting drug development in rare cancers, the Rare Cancers Program leverages multiple OCE projects and initiatives to address the challenges of developing new treatments that affect small numbers of patients. This also includes collaboration with stakeholders both inside and outside of FDA to identify opportunities to decrease obstacles, better harness scientific knowledge, and strengthen coordination to facilitate efficient drug development. Next slide, please. Project Catalyst is intended to foster early-stage oncology product innovation and development. It does this through scientific discussion, education, guidance, and regulatory engagement with a focus on academic life science incubators and accelerators, as well as small pharmaceutical companies. The guidance and educational resources provided by Project Catalyst help support informed anti-cancer therapy development to expedite the availability of cancer treatments to the public. So examples of this include facilitating speed dating-like meetings, allowing investigators to ask questions regarding their drug development plans at an early stage. Another example is the development of the Oncology Regulatory Expertise and Early Guidance Program, or OREG. This is a self-directed platform providing early-stage oncology companies with product type advice to inform sound product-specific drug development decisions. Next slide, please. So we all know that traditional, prospective, randomized controlled trials in oncology can be burdensome to trial participants, investigators, and trial sponsors. And particularly in rare cancers, they're not always feasible. So Project Pragmatica seeks to introduce functional efficiencies and enhance patient centricity by integrating aspects of clinical trials with real-world routine clinical practice through the appropriate use of pragmatic design elements. An example of this is the Pragmatica Lung Cancer Treatment Trial, which is an NCI phase 3 clinical trial for patients with metastatic non-small cell lung cancer, randomized to receive standard chemotherapy or a combination of ramucirumab and pembrolizumab, with a single primary endpoint of survival. So this trial is designed to remove many of the barriers that prevent patients from joining clinical trials, such as allowing a lower performance score and having fewer assessments and visits. This was a major OCE effort with collaboration between FDA, NCI, and industry. Next slide, please. We also discussed challenges such as endpoint selection and lack of controls. The Oncology Real World Evidence Program seeks to engage in evidence development modernization to advance the application of real-world data to generate real-world evidence for regulatory purposes. The appropriate use of real-world evidence may aid in better characterizing the natural history of rare cancers, informing trial design, and enhancing trial efficiency. Next slide, please. I'll just briefly mention Project Significant, which aims to promote collaboration and engagement to further the design and analysis of cancer clinical trials, and this is particularly important for rare cancers like sarcoma. Next slide, please. While not a particular OCE program, tissue-agnostic initiatives may benefit sarcoma drug development by including sarcomas in larger studies along with other tumor types. Various cancers, as we know, may share a common targetable biomarker. And examples of this, as mentioned previously, are RET fusion-positive tumors, NTRK fusion-positive tumors, and MSI-high DMMR cancers. There is a key difference between traditional oncology drug development and tissue-agnostic oncology drug development, and that is the need to generalize treatment effects based on responses observed in some cancer types harboring a specific molecular alteration to other cancer types with the same molecular alteration, some of which may not have been studied in clinical trials. So if we accept the tissue-agnostic approach as being valid, then we accept the principle of generalization along with the degree of uncertainty that comes with it. Next slide, please. The last program I will discuss is Project Orbis, which facilitates the submission and review of oncology marketing applications across the participating countries. This is a collaborative review program launched in 2019 to provide a framework for concurrent submission and review of oncology products among international partners. This may allow patients with cancer to receive earlier access to products in other countries where there may be significant delays in regulatory submissions. It is important to note that each country retains its independent decision-making for each application. And of note, it was recently announced that the EMA has become an observer of Project Orbis. Next slide, please. So given the importance of international engagement and sponsor interaction to furthering drug development for sarcomas, FDA has several mechanisms for international collaboration, and I've provided just a partial list on this slide. We particularly value opportunities to engage with the EMA, and these regular interactions with EMA related to cancer drug development began in 2004, and we now have monthly meetings with EMA and 4 additional regulatory authorities to discuss international cancer drug development programs. Specific to pediatrics, given all pediatric cancers considered rare, the Accelerate platform brings together academia, industry, advocacy, and regulators, including engagement with international regulatory environments, to find solutions for more and better innovative therapies for children and adolescents with cancer. Efforts include organization of multi-stakeholder pediatric strategy forums and conferences to discuss strategies for bringing therapies to pediatric cancer patients. Mini symposia are small stakeholder meetings designed to discuss specific topics of interest that are open to international engagement. And pertinent to today's discussion, I'll give an example of a mini symposium held in 2023 pertaining to osteosarcoma. And lastly, Conversations on Cancer is an OCE public panel discussion series examining cancer-related social issues with inclusive and diverse panel members. And October's meeting, which was focused on metastatic breast cancer, was a collaboration of FDA and EMA. Next slide, please. So in summary, I think we're all aware of the many challenges to the development of drugs for rare cancers, including sarcomas. Today, I tried to provide a high-level overview of ways in which FDA, and particularly OCE, are combating these challenges with projects and initiatives aimed at using collaboration both inside and outside the agency to bring safe and effective therapies to patients with rare cancers. We welcome opportunities to engage with stakeholders early and often to facilitate new treatments for patients with sarcoma. Next slide, please. I'd like to leave you with some resources highlighting FDA guidances relevant to sarcoma drug development and rare cancer drug development. Next slide, please. As well as the website and email for the Rare Cancers Program, should anyone want more information or have any questions regarding future involvement. I'd like to thank my colleagues within the OCE, particularly Martha Donahue, Leslie Doros, and Mark Nielsen, and I'm looking forward to the rest of today's discussion. Thank you so much.
Thank you, Caitlin. And I would say that your slide for the conclusion, I could replace FDA by EMA, or even better, I would add EMA to FDA. We are exactly on the same line here. We need more engagement with the stakeholders, more frequent and less formalistic, I would add, processes to discuss and to progress rapidly. Because when something is very difficult, better meeting often than avoiding the meetings just to pretend that the difficulty doesn't exist. So we will discuss now in a round table the questions that came from the presentations, and we have tried to list these questions, thanks Caroline, into two groups of questions. The first group is on the endpoints, the research questions to use to establish efficacy. And I would say that these questions come from the investigators and should be answered by the regulators, probably. The second group is the questions on the study design to use. And here, I believe that it is a discussion for anyone. We all have to address this question together. And the last one is how to create a framework for developing rare cancer medicines. This is the totality of audience, but some proposals should come from the regulators. And you will see that I would have a proposal for a simple conclusion in 3 bullet points, but this conclusion is opening many doors. I hope so. So if we go back for the first one, and I would group the questions to one single question. What is the best endpoint? What do regulators expect as a demonstration for a clinical benefit that could be used in a benefit-risk balance assessment? And ancillary question, but important, when tumors do not shrink under treatment, how could we evidence that the medicine is active without any shrinkage, in other terms, without any response rate according to RECIST criteria? I don't know. Maybe I could try to answer as the chair of the Oncology Working Party, maybe, but under the control of my colleagues, Oli, the vice chair here, Francesco, and all the colleagues from the side of the EMA. The best endpoint, there is no one best endpoint, but something which is normally highly reliable when you cannot randomize. If you randomize, obviously the endpoints are very classical. We prefer overall survival, but when it is impossible, progression-free survival and so on, but you probably know the notable guidance. When we cannot control, the most reliable endpoint to solve the problems due to the absence of randomization that normally solve the imbalances at baseline is response rate. I just explained why it is our favorite endpoint, because normally you do not expect a severe tumor to shrink spontaneously. So any shrinkage is explained by the activity of the drug, and this is a starting point. We normally do not expect benefit without shrinkage. So the response rate is the proof, not so much of efficacy, but of activity. Then we have to deal with the clinical significance of a response rate. And then comes the question of how much should be the response rate to be the magic threshold that results into a conclusion of a benefit. We do not have a simple answer to that. First point, the duration of response is of importance, but this is a time-related endpoint, which is sensitive to the baseline factors as well. But you could say that for every patient having responded to the treatment, we get a bit rid of this idea of the baseline characteristics. So the duration of response is there just to check that we do not have many responses, but of no significance because transient responses that wouldn't bring anything to the patient. A drug that puts 20%, 40%, or 90% of the patients under responses, but the responses last for 1 month is probably not very useful. There is the formal test to compare here. Okay, it must be pre-planned, but it's not as reliable as the one for the response rate as a crude number. Then come the other endpoints, PFS, overall survival, that would support the conclusions and are very important. Because if we do not see any difference, or even less, if we see a detriment, we have to explain that. And if a drug elicits many responses, but we do not see any difference in PFS and overall survival, this challenges a bit the idea on the response rate. And even more, if we have a trend to a negative effect, it would mean that you offer the patient a drug that is active on the disease, but that kills the patient. So just to make an extreme. So to summarize, this is in the guideline, however, we believe that when you have no comparator or an external comparator, overall response rate is the favorite endpoint. And you could see that some approvals based on single-arm trials in oncology, most of them were based on response rate, which is normally not our favorite endpoint, with the support of duration of response, PFS, and overall survival. To tell that in other terms, activity is great, provided that toxicity is not destroying the benefit obtained through the activity. In the question, when you do not obtain tumor shrinkage, how could you evidence the benefit? The answer, in my opinion, is relatively simple. If you do not obtain any shrinkage, and if spontaneously tumors never shrink spontaneously, but do not grow up that rapidly, this is a failure. No, but the difference is when you can establish through the natural history of the disease, and once more, this is where the registries, the observation of the natural history and so on, is that important. If you can demonstrate that tumors in that disease inevitably progress with a kinetics that is highly predictable, there, the difference is of the same nature as the difference between tumors that do not shrink and tumors that shrink. Tumors that normally progress inevitably without any example of tumors remaining stable. If you obtain stability, I believe that you demonstrated the activity at least. And you are even in a relatively good position because normally the progression of the tumor is associated with more symptoms, with more severe disease and with more severe outcomes. But this is a bit more difficult because you have to trust on the demonstration obtained from an external body of data. So to summarize, ORR is the favorite endpoint because it is the most reliable, but it is not that related to the clinical benefit, and it has to be supported by an impression of an acceptable toxicity and of a clinical benefit to the patient, whichever demonstration, knowing that the solid, robust demonstration on criteria such as PFS or overall survival, it is difficult to establish when you have no randomization, except if the difference is that large that you must believe in it. But we are here under a best case scenario that doesn't happen that frequently. So it will be my answer, but I would be happy to Francesco, to Elias, to Oli, if you want to comment on top of what I just said.
Yeah, if I can just add to this, and perhaps going back to what Hugh was asking in his presentation, that we would like clarity here, and we would all wish that there was some intrinsic quantity that endpoints reflected that we could just refer to and quote the cutoff and agree on that cutoff of that intrinsic quantity, and then we would have our answer. But I think this is an illusion that there is such a thing. All we have is a number of endpoints, and rather than striving for the optimal endpoint, which perhaps we can all agree what is the ideal endpoint, we are more like mud diggers trying to rescue anything that is meaningful enough for a decision. And this is unfortunately not something that we can resolve through a tick box approach or a one size fits all, but we need to always look at all the evidence in relation to each other. And this is not only related to the endpoints, but the endpoints are only as good as they are able to help us in making a decision. And then in the decision, it's not just about the endpoints, it's also about our risk attitudes. How worried are we to make mistakes or not? So that's why a crystal clear answer cannot be given. But I think we need to continue talking to each other, continue to make sure that we communicate how we value endpoints, how patients value an improvement in PFS, and when these improvements in single-arm trials or in randomized trials are such that they allow comfortable decisions. I think that's the discussion we need to have, rather than hoping that we will all find the single measure that will allow us to mechanically take decision. I think that's an illusion. But let's continue talking. Let's continue to develop a common understanding. Regulators do not have any special wisdom to decide what is a good endpoint or not. Patients do. Clinicians who listen to patients perhaps more than regulators, more frequently than regulators, they do. So that's the communication we want to develop.
Yeah, I think the issue of clarity would be wonderful, but I completely agree that it's not always achievable. And we won't be able to define a set of endpoints that apply to every sarcoma because they're all different and the data sets and the research is set. But I want to go back to something you said right at the beginning, Pierre, which was that one of the objectives of globally of what we're trying to achieve here is to close the gap between the regulator and the clinicians. And I think in the case of EHE, it's a great example because from a patient perspective, what we see is highly experienced clinicians, Dr. Stacchiotti, Professor Jones, Bill Tapp in America, who are now prescribing sirolimus for EHE. They don't do that frivolously. It's taken many years of gently moving to get to that point. And they now all agree, I think, in the right cases, not for all EHE patients, but for right patients, that the frontline drug, first-line drug is sirolimus. And in each case, I think they have it subconsciously or consciously in their own minds identified the endpoint, and they've compared sirolimus to that, and they've said, this is the right drug. And so that's the gap we need to close. I think we need clinicians and regulators to come together. And what would be great was for the regulators to say, right, you are the guys prescribing the drug. You are the guys with the clinical expertise. And then for every other treatment that the clinicians prescribe, our national health systems put the responsibility for that decision on their shoulders. Whether it's no treatment, and that's a big decision in EHE, to go to wait and watch, whether it's resection or transplantation or embolization or any of the others or systemic treatment, we trust the clinicians. We require the clinicians to make that decision. And so From a patient perspective, we see clinicians prescribing sirolimus, or patients do, and they think, why is that not acceptable? And I'm not saying it, you're not saying it's not acceptable, but it's an area of understanding where we would love people to come together and for you to say, explain to us clinicians, how have you reached this decision? And then we would challenge you to say, okay, what's wrong with that? Why can you not accept that? That would be a fantastic conversation to have, I think.
Picking up a theme here, which is everybody in this room is agreeing that regular discussions are essential to plug those gaps, to discuss individual questions. And we've had many questions that, that have arisen, and they're all listed there. Could we not commit to having regular scheduled meetings where we can come together? Because if we do this on an ad hoc basis for this case or that case, it's going to take forever. There are so many questions, so many diseases. It would be a real plus, a real benefit if the outcome of today is this is the schedule of meetings and these are the questions that we will discuss on these meetings. I think that would be a real win for the clinicians, for the patients, and for the regulators.
I do agree. I do agree, and I believe that one of the conclusions I would propose is how could we make the interaction between all the stakeholders here more flexible, more adaptive, and how could we find solutions, forgive me, to break the rules, which are not necessarily protecting the system, the patients, the prescribers, and the regulators. Just to give you an example, we have the scientific advices. Scientific advices are a great process. As the vice chair of the scientific advisory party, I would never say anything else, because if I wouldn't trust the system, I wouldn't have applied to be the vice chair. But okay, nevertheless, it's not the solution to anything, to everything. It's the solution for classical developments with specific questions related to one drug, to one product, or to one process to develop new methods and so on. But in the case we are just discussing, it's not necessarily the best frame. We had the experience of that during our discussions around EHE. The idea was to have this as similar as the scientific advice process. Hopefully, great news, it was a pilot, so we were relatively free. And we decided to break the system. Having the conclusion within 2 months was a nonsense. Not taking time to control how we can modify few things to meet the regulatory requirements takes much longer. Having a formal discussion during a discussion meeting of 90 minutes here in Amsterdam was not the appropriate solution. And we decided that we would discuss as soon as needed to follow the developments of the question till we find a solution, which is not exactly what is performed by the scientific advices. So this is possible. This could become, we are addressing the one part of the third question here. This is one part of that solution. Let's be creative. So we discussed with Francesco on possible solutions during the coffee break. As I told you, we discussed yesterday with Paolo Foggi, the chair of the SOAP, both of us, solutions to improve the system for scientific advices that are not normal, if I could say so, scientific advices. How could make the system more efficient, more flexible for the benefit of anyone. So one commitment would be, and it is one of the conclusions I proposed in advance, one commitment would be from us regulators to reflect on how we could not build new rigid systems, how we could build adaptive systems to meet as soon as necessary to solve the problems as soon as we see them appearing. whichever the process that will give birth to that one. Should it be the scientific advices? Should it be consultations of the oncology working party, of any other body? Who cares? Who cares? Provided that it works. So from our side, be sure that we will start next week reflecting on how we can modify the processes. And in the meanwhile, if we need, and EHE was a good example, if we need more meetings, if we need to adapt the process, we will do so. And if we put a name on the process, but not really necessary. We need rules, but we are not keeping the rules. The rules are keeping us, which is different.
Can I add something about the endpoints and the questions that are pointed here in relation to what you're saying? I think if you speak about rare cancers, well, we already established that's a wide variety of cancers and they all behave differently. So even endpoints can be different for different cancers, right? Because, and I give you examples, right? EHE can be very indolent and can be stable for a long time, and patients might not need treatment. Some of them can be very progressive, and if you have a drug that stops it from progressing for years and years, I would think, stable disease is a good endpoint for EHE. Another example is I'm part of neoadjuvant immunotherapy trials in melanoma, and then you have many positive macroscopic nodes, and you give 2 cycles of ipi/nivo, and what happens? No recess response. Then we take out 1 lymph node, complete pathological response. And these patients do very well, so there's no shrinkage of the tumor, but you take something out and you see a pathological response. And, we have actually proved with a single arm trial that it is correlating with outcome. So, so I'm saying different endpoints, even if the tumor doesn't shrink. I don't think shrinkage is always the best endpoint. We have GISTs also in the sarcoma world. We call it Choi response. The tumors become cystic, they don't shrink. So if you use shrinkage as a rule, then, you will never have that drug available. So I think if you deal with many different types of rare cancers, endpoints should be able to be different depending on biology of the disease if we explain it well.
Exactly. And I believe adjuvant, neoadjuvant is a great example. And the cure rate here might be a very good endpoint. So apparently the disease disappeared after surgery. So where is the response? Nowhere. So, but if you see that 50% of the patients haven't relapsed after 1 year, when the natural history of the disease tells you that inevitably after 6 months, you observe relapse despite complete surgery, then you have your endpoint. So we must be creative. I completely agree with you.
And one addition, if these patients have, for example, 10 positive nodes, we take 1, and you see a complete response, we leave the other 9. And nothing changes. It's a complete response, right? Yeah. But it's fibrotic. So it doesn't, so that's what I'm saying. If you have something you take out as an endpoint, pathological response, then... Yeah.
So just to follow up with your remark, the tumor shrinkage in a metastatic setting may be also very confusing because we have pseudo progression with some drug. Although it's effective, we are unable to interpret. And that's the debate we have in sarcoma versus RECIST Choi criteria. That means that we will have to be very cautious with this.
Just to be clear, I never said that overall response rate is always the best endpoint. I just said that activity is the most reliable endpoint to start the assessment. And if this activity is difficult to establish, normally it's relatively simple, but in some situations, pseudo progression, adjuvant, neoadjuvant setting, where we do not see anything, or tumors that normally progress and there remain stable. So it is not response, but it is a proof of activity. Yes, we must be creative. Obviously, but my point was that we must refer to the natural history of the disease, and the best endpoint to start the demonstration of the benefit is an endpoint that is as independent as possible from the baseline prognostic factors that could be even ill distributed because the absence of randomization. Then comes the chain Francesco alluded to of other arguments. This is the reason why, let's go back to overall response rate. How much? 30%, 35%, 40%? Obviously, 100% is something, 0% is nothing. But in between, I don't know. In some tumors, getting 20% response rate is marvelous because it was completely unexpected. If the drug is devoid of any toxicity, it's perfect. Sometimes 60% is not that interesting. So the magic number doesn't exist because the natural history of the disease is very important and because the other endpoints that are not as demonstrative but very supportive are important as well. So if you observe a limited response rate but an impressive PFS, even if PFS is not very reliable, this is convincing.
I think Bill Tapp from Memorial raised his hand already a long time ago. So okay.
Thank you. Are you able to hear me at all?
Yes, Bill.
Oh, great. Thank you. So first of all, I just want to say this has been an absolutely wonderful meeting, and I appreciate everyone coming together and have this discussion. Interestingly, when I look at it, I've seen a tremendous shift from an academic standpoint on how we want to think about caring for our patients. And it's really become directive on our side from an action standpoint, but also very collaborative. It's the first time in 20 years I've seen such a level of collaboration. And importantly, it's been really patient-centric and inclusive. Hence we're doing everything with the patients at our side. And I hear the same from the regulatory bodies. I think this is a wonderful opportunity we now have to think about how to collaborate and shift paradigms for treatments for our patients. And one of the things that I think is interesting when we look at these questions, sarcoma really represents an opportunity because we deal with so many different rare diseases. So can we think about moving forward how to set paradigms for rare cancers, right? As opposed to thinking of one sarcoma subtype. The other thing that's interesting is even as experts, we don't really understand a lot of these diseases. Many of them are newly identified, genomically based, and unless we have a very distinct target, we're struggling to understand how it affects patients, what's the biology, and how to develop drugs. And so, it's often difficult to come to regulators with a very concrete plan on what needs to be done. And so one of the things that's I'm seeing from a lot of different places is this massive collection of data so we can better understand the diseases, the natural histories, the response patterns. And I wonder too, if we think about dialogue, if we think about this collaboration, could we set up practical frameworks to it where we can discuss data collection, where we can discuss diseases, where we can discuss what we're finding and then use our collaborative hats and expertise to say, how can we shape that, in the various aspects of these diseases we're encountering to say, how can we change or shift drug development paradigms for, right? So, maybe think of a broader blueprint to say how this collaboration can work so that we can really bring everyone's expertise on the table as we're defining, I think, some of these diseases and what we need to do.
Thank you. And thank you really, Pierre, because it's the first time that I can read between words the attempt to really go beyond the rules. Actually, I would follow up with what Pan and also Bill just said, because, and I think also Raf this morning said that EMA can join the project. Today, Lorenzo presented Push It, which, as Sylvia said, it's just a part of Push. So we are here with our project. So why don't we think, as you are also considering, the way we can work together, to take as a framework, Bill, the idea of Push and Push It, because this will allow us to basically work together on different type of information sources, different type of studies that could then be complemented each other. And we might, and this is also from the expert position, we might identify maybe 2 or 3 use cases in terms of specific diseases with different characteristics, so that we can test accordingly to 3 different models, the type of data and the type of studies that could be done within a framework that has been already thought, as it was a project asking EMA and FDA to contribute to define, okay, some models on specific use case, if this could be an option, so that, you know, in the different meetings, we have specific use case and data sources to consider. Just an idea.
So my answer would be a question, why not? No, absolutely, why not?
Yeah, not if it's for product development directly, but we don't know enough about this. At least I don't, and so that's interesting. But there are. This is why not everything maybe was so very clear. There are some considerations. It's about the how. I think it's flexible enough. I would suggest that a starting point is how to structure an agenda. And I'm saying this because I feel that deeper dives are necessary into the matter. Pierre, you gave an answer to a simple question that showed how the question is difficult, and Francesco, you elaborated the context. So a deeper dive is necessary. We don't have a monopoly for organizing meetings and bringing stakeholders together, and I'm very happy how Accelerated presented how they have done it for 6 years, and I really strongly recommend to consider that as a model. But for the agenda, to structure it, I think I see the endpoint questions as difficult ones that require continual and case-based and general exchange, etc. So they should be started, but they will maybe not lead to the first conclusions. I think what could also be started early are discussions about registry, because Silvia, you mentioned these are projects that are starting up right now, and you are interested to obtain input. And also the platform considerations, I believe, could be fed into one of the several ways of discussion that we have offered. And of course, we will help you in finding the right forum and way of discussing it. So these endpoint registry and platform for me could be topics for deeper dives. And I think eventually the endpoint discussion will need to be coupled with medicines, because that is too abstract and too general to hang in the air. Other topics will then be secondary, like number of patients, interim analysis, how to move away from last line into earlier lines of treatment, but they are important from a public health perspective. So I suggest that instead of the format, we think about the agenda that would structure tackling the issues at hand.
Important. I just had Martha Donahue waiting for the floor, and then it will be your turns. No. Martha? Martha, we cannot hear you. Are you still there?
I think you're muted, Martha.
So maybe while Martha is joining, I think that all of this would be absolutely great. And if this was the output from this meeting that we were really wishing, expecting, desiring, I think that as we are thinking of a global collaboration, would be great that also FDA can maybe take part to this kind of meeting and discussion, so that we can have, as we had today, the two perspectives. So I don't know if Martha, that is maybe now joining. Okay. Oh, okay. So in the meantime, maybe Vinette, yes.
I hope you can hear me. Yeah. So this was a wonderful discussion, and I think it points at very important issues, and I'm really happy about the patient's input today. When we will talk with EMA, I think it's important to realize we are not only for sarcoma here. although that's more tangible for us because we are sarcoma specialists. And I'm talking also from the perspective of the EORTC, where we, of course, handle with a lot of different tumor types, where the issue of differences in endpoints and also biology of the tumor is important. But where also patients have different inputs, and patients are also different because some tumors appear at young age, and we heard a lot about pediatric oncology, and all others are elderly, and also that impacts a lot of the endpoints of studies, because we all know that elderly patients appreciate more quality of life than length of life, to say it a bit black and white, than children do. So I would strongly suggest also to have an input on quality of life. And also in the rare cancer field, we know that, for example, EORTC has a lot of, in the development of quality of life analysis of patients with rare tumors at the moment. So that may be helpful for the discussion. We always start with biology and molecular markers and how to treat patients, but we can also make patients really worse due to all these drugs. So I think the whole picture and the whole, I would say, organizations in Europe and across the world who have their own background and can deliver data are important if we have these discussions. So I would say to plea a bit for a broader view on these aspects. And I think that will be helpful because it's not easy where we are talking about today.
Sure. Complete agreements. First announcement, the 29th of February, we're having a workshop on quality of life in cancer. So probably we will have an opportunity to discuss this point a bit further. Second point, you're completely right. And something I would see as very important is that during studies, longitudinal studies, investigating many elements of on patients with a given condition, disease, and so on, it would be great if we have investigation on quality of life, not within a clinical trial, but just to understand what matters much to the patients. Because, as you said, the perception of a given cancer with a given prognosis could be completely different in children and in the elderly. It could be completely different depending on whether the disease is painful or not. It's completely different as to according to whether it is affecting the central nervous system or not. I could have many examples for that. So understanding what matters to the patients with the help of the patients as well is very important, because when we make a final judgment of the benefit risk, if we see an improvement in a parameter which is cardinal to the patients to get some help from the treatment, it will be much more convincing. So I completely agree with you. And this is an important parameter we should not neglect. Martha has joined. Sorry, just wait a second. Martha, please.
Hi, hopefully, can you hear me now?
Perfectly.
Oh, great. Thank you for your patience. And thank you so much for inviting FDA to participate in this wonderful meeting. For those of you who don't know me, I'm Martha Donahue. I'm a pediatric oncologist by training, and I'm currently serving as the Associate Director for Pediatric Oncology and Rare Cancers in the Oncology Center of Excellence at the FDA. I think this discussion has been really, really interesting. And I guess I just wanted to provide my perspective in terms of some of the topics that have been touched upon already. And, I completely agree that there is no one-size-fits-all response, criteria or efficacy endpoint, I think, across sarcomas or even across cancers in general. And that, I think, the approach that we try to take is to really tailor our expectation in terms of efficacy endpoint, to one that we think could be clinically meaningful, but also one that is relevant and feasible for a specific disease. And sometimes it's not a single endpoint. Sometimes it's a combination of endpoints that really help us understand effectiveness. Not to say that we have the answer for every case, but I think having prospective discussion, with us in conjunction with EMA can I think really help all of us align on the plan that would best suit a particular development program for a specific sarcoma subtype. In terms of what degree of response is clinically meaningful, I think that does vary depending upon the situation. Obviously, we'd love higher response rates, but even a more moderate, lower response rate, if accompanied by clinical outcome assessment, measures that demonstrate that patients are benefiting from treatment, or something that can be really important to help establish effectiveness. I think Dr. Wiedemann, we've had experience, with the selumetinib development program for plexiform neurofibroma, where the response rate was modest, but when combined with some clinical outcome assessment measures, really helped us understand the benefit of that drug for patients. And so I think we would appreciate being brought in as early and as often, to these discussions. If there's a framework that's established, or a series of meetings that are established, we'd be, with EMA and the rest of the stakeholder community, we'd be really interested in participating as well. We are definitely not the experts in, what's the best endpoint, but we do, have a broad understanding of the sarcoma development in general. We certainly have familiarity with a variety of endpoints, and can bring, and communicate sort of what, provide some advice that may help streamline development programs. And, things such as use of real world evidence can be, certainly things that we can entertain using, but it's also a very highly complex topic that, we would definitely like to be part of early. So I'll stop there because I don't want to take up all the oxygen, but just wanted to, again, thank you for having us, and hopefully we can continue to work together.
You're more than welcome. Maybe your comment is exactly the right one at that stage. Discussing any further the merits of a given endpoint, the quantity and so on, is it really useful today? It might be a discussion which is specific of any question, any disease, any drug, any development. So it is something we have to discuss, for sure. The principles are, in general, by oversimplification, what I answered at the beginning of this bullet point on the endpoint. But I think that, yes, you're right. Let's consult. And if it is a joint consultation with the EMA and the FDA, even better, because the more we are, the more we could be clever. So I think the collaboration is very important, and not giving diverging responses, not likely, but it could happen, is better. So I have two things. Are there last interventions on this question of the endpoint, given that we have room to discuss for each development, so maybe let's not extend too much. And if yes, please. And I would like to give the floor as well to Wim Ooyen to bring his point of view, if necessary, on that question, because he is an expert in some methods to assess some endpoints that are of importance. So questions? No. So Wim, if you want.
Yeah, I've been listening carefully. Obviously, I'm not a sarcoma specialist, but I'm an imaging specialist. And so when I listen to what I hear, I want to look at the analogy of what happened with molecular imaging in lymphoma. And many years ago, it was considered complicated, difficult to implement. And actually, EORTC was one of the first to run a very large trial that really changed the perspective of using advanced imaging as a surrogate marker. And subsequent trials resulted in better patient stratification, taking away toxic drugs that were not needed, looking at patients who were imminent to fail therapy, where early treatment intensification was considered. And now it's basically the mainstream of imaging. And it all started with the H10 trial at EORTC. And it took mainly academic efforts to get those methodology into clinical mainstream. So let's say I would advocate also to look at, let's say, other than traditional methodology to incorporate into clinical studies, let's say, not just exploratory and underpowered as usual, but also to have the courage to get it adequately powered site study so that you can make a point to incorporate it as new endpoints in subsequent clinical trials.
Yes, very important remark. Thanks a lot. Yes, it's not only the endpoint per se or quantity that matters. It is the way you measure it, and maybe with the methods you could find different endpoints that could be more relevant to a given disease. So once more, it means that whichever the development, the discussion around the endpoint is critical, and better discussing that in advance with the regulators, even better if we can discuss during joint meetings with our FDA colleagues. It will be more than welcome. Once more, we are open to discussion. We must find models to have these discussions organized, but this shouldn't be a problem. Shouldn't be a problem. Yes, please.
No, thank you. I love the idea of this, but for many of these ultra rare tumors, we have one shot at doing any sort of prospective study. It'll take us 10 years to get enough patients, and we don't have time or number of patients to validate a new method. So we can do it in a different disease and try to extend it to these rare sarcomas, but as we're talking, they behave differently and they have different outcome results. So I love the idea of developing alternative methods. It's really hard to think about that in a disease where there might be 50 patients in the world that have it. And how do we design endpoints at the same time that we're doing the prospective study to try to show benefit of a drug? So that's part of the challenge. It's different. You said a very large study in lymphoma that did this. We'll never have a very large study in any sarcoma.
No, I think I fully appreciate, let's say, the difference between rare and ultra rare, but let's say lymphoma is a rare disease. And if you look at the subtypes, for example, in non-Hodgkin lymphoma, which there is a huge spectrum of different subtypes that behave totally different. Let's say still, let's say taking it from the Hodgkin trials where it all started, let's say it is progressing and you see that the community is picking it up. And let's say you very eloquently made the remark, what if tumors don't shrink? Mm. And say, if you look at sarcoma, for example, there are new targeting agents around that we just starting to understand, like the FAPI, which is probably in sarcoma a much more attractive agent than the traditional FDG. But let's say if we keep on identifying the hurdles of novel technology to work towards endpoints, we'll never get there. And it has to start somewhere with the observational studies, like in sarcoma. I'm not advocating at all that you put a new technology into a prospective clinical trial and bombard it into an endpoint. But let's say if we don't start even yet observational studies, we end up nowhere.
Agreed.
So if I may, we spent 40 minutes out of 60 on discussing the endpoints. And I'm a bit nervous because I believe that most of that discussion should happen later on. So we are not here to start the specific meetings. We are here to discuss how we can improve the process. So I saw a question, except it is a very burning question, not in the detail on the endpoints, please, but I would like we stop on this topic, except it's very important.
Actually, I'm bridging on the second point.
So that's marvelous.
Yeah. So, I mean, for me, one of the a very practical question to the regulators is about the way we measure the endpoints in terms of which is the data you really want to see. We have heard about registries or observational studies that are not exactly the same things. There are also prospectively maintained databases that usually are used by clinicians to actually support their observations that are also sometimes valuable, sometimes less valuable. I think it's very important to understand which is the minimum quality of data that you want to see in a retrospective fashion and prospective fashion to substitute for randomized data that will never be or will likely not be available in many of these ultra-rare diseases. So that's for me is a very practical question that I think is important to make, and it has to do with also endpoints when it comes to understand how often you need to measure things to say what is going on, and to harmonize this among the different data sets that you're actually putting together, and how you want this to be monitored if a monitor is needed for understanding your data sets. I mean, the quality of the data sets and hence the quality of the observation on the endpoint you have decided up front.
So my answer will be simple and short. This depends on the development, which is the topic for discussion. This depends on the drug, on the disease, on stage. So answering here and now would be highly premature, I'm afraid. So no magic response, I'm sorry.
Yes, but yet we are talking about mainly here, we are talking about the development of drugs in the advanced setting. We are not talking of other stuff. So basically we are here discussing a situation where you need a drug and the drug is not available and the patient has metastatic disease. So that's already a specific scenario compared to other adjuvant, neoadjuvant setting or stuff like that. Because
yes, the topic is not as large as the topic of oncology in general, but it remains large enough and valuable enough to prevent me from giving you an answer at that stage. I'm sorry. So, yeah.
I think we're very active in this field, exploring, how to move forward, what are the opportunities and so on. Workshops have been mentioned and guidelines and so on. And again, it's not going to be a one-size-fits-all probably type of solution in the end, but there will be situations, many believe, where there are huge opportunities from that, and others where this is hopeless. There was a study that we did a few years ago where we showed that actually some degree of impact of real-world data has been quite frequent in many applications assessed by the EMA. So it's also about the role of these data. But many in the system are open and optimistic about this. Others are not, and the jury is still out. But
so maybe there is Robin.
Yeah, so I come back to the point that I made this morning and that Alex has essentially made now about high quality data. I think the crux of this is the definition of high quality data. And don't get me wrong, I think it's great that we're having this discussion. And I don't want to seem, combative about this, but it's really important that this is dealt with, that we define what you mean by high quality data, because I think for if a drug has a 90% response rate that's durable, then a single arm trial is going to show that. But if something is more marginal, it's very difficult to tease out the benefit of a drug, and trying to use endpoints such as response rate, resist response, or progression-free survival, comparing to retrospective data when the selection of target lesions, the timing of scans can vary. It's a real challenge, and I think the crux of this is trying to define what the regulators want in terms of high quality real world data. And I'm sorry to sort of go on about this again, but I think that's really the elephant in the room here.
So the answer is not, we will refuse anything that is not high quality under the definition which is listed below. No, we are not that tough. I would say that the ideal quality of data is standardized, reliable, robust data. So in other terms, if you come with an evidence where all the patients have been investigated according to similar schedules with the same methods, measuring the response the same way and so on, it is high quality data probably. If unfortunately you cannot do that, and if the schedule of assessment of the response varies across the centers and so on, we will have to make a decision, however, but it will be much more difficult. So the answer to your question is there is no a definition of high quality data as opposed to bad quality data. I cannot do that. But I could tell you what is common sense, that standardized, recognized methods as controlled as possible with quality insurance and so on, is high quality data. I cannot tell anything, but I believe you know that better than me. So, Brigitte, so I'm sorry, the conclusions will be restricted to the endpoints. Not my fault. Not my fault. But if you want to discuss that only, feel free. But okay. So Brigitte and 3 other. So I'm desperate. So Brigitte, please.
Yeah, this is just a quick comment to high quality data, which I think is so important, especially if we think of non-randomized trials and external control. And I actually wanted to ask, I thought the effort with PUSH-IT and also the registry are so amazing, but I think what this means is that the agencies would have to be able to audit this data and find the source document. And I was, for each of the evaluations or, whatever endpoints are measured. And my question would be, especially for longitudinal studies or more slowly growing tumors, such as maybe GIST or chordoma, is this being done with PUSH-IT right now or with the EORTC? And what funding would be needed? And would it maybe make sense to explore the development of an external control with this more strict guidelines for diseases where an interventional trial is planned, so that one upfront with the regulatory agencies could decide these are the things that are needed? In my experience with my part, it is difficult to collect this, even in a study where there is funding. Thank you.
I gave up. Somebody else asked for the floor. So I told you.
Yes, I'm Gloria Marquina, and my question is about the registries, and it has something to do with that the person has just spoken about, the funding. But the thing is, as you have said, you have not have any definition for high quality data of the registries, but we are all putting data on the registries, and well, there is lack of funding, so we are manually entering the data at the moment. If you are saying, and this has to do with the endpoints, if you are saying that if it is something heterogeneous, for example, the measurements of the CT scans, for example, the schedule of their CT scan, should we, as a community, as the sarcoma community, ask our colleagues that are collaborating in those databases, ask them to do the same measurements for the patients? Should we do something like that in order to avoid that heterogeneity? Thank you.
So I'm sorry, we are entering discussions where anyone is offering his own development and asking questions for his or her own development. This is probably not the right place. We have a guideline on registries, so you could follow without any problem. If we engage into specific discussions on each project, it is exactly what I tried to avoid with my introductory statements. So we have now a few minutes to deal with the 2 remaining points. Because I have to leave at 5 sharp, I would suggest that we use 5 minutes. Sorry for that. 5 minutes for the 2 following bullet points, and 5 minutes for the conclusion. I'm really sorry. So The second bullet point was the research question and the study design to use, but I believe we addressed indirectly some of these questions together. I would say that this is normally up to you to answer with our help. And this would be topic for these informal discussions we would like to set up. And indeed, we touched upon the quality of the data. As a regulator, I believe that we don't have a strict definition of the quality of the data, but better is better than good. So this is the only answer I have. Collaboration is something which in rare disease is absolutely crucial. When we say collaboration, we have to deal with possible heterogeneity. There is nothing we could do to avoid that completely, but we should try to control for that as much as we can. So, and for the clinical trials, the observations, I would express exactly the same remarks, but feel free to comment if you want. Once more, we could offer, we will do our best to offer fora to discuss that with you point by point, development by development. So please, in your actions, don't begin with, but with my registry, with my trial, I would like if I can, no. Just general remarks on that, if you can, please.
Just a general remark, a question to the EMA. So we were talking sometimes about drugs that are off patent, and is the EMA the right place then to go to? Of course, we need the registries there, but there could be an alternative route with ESMO. Is EMA interested in? We have imatinib for chordoma. We have heard about sirolimus for EHE. These are drugs that are off patent. The companies are not interested anymore. Should we go to you or should we go elsewhere?
Not only we are interested, but we are offering a specific process with this repurposing process. So if you have a drug which is used for other conditions, but you believe it could be very useful for patients you treat, which are an ultra rare condition, repurposing is exactly the process we could propose, with the limits that we observed this morning when we discussed that we need somebody in the end to take the responsibility for putting the drug on the market and taking the responsibility of pharmacovigilance and so on. But there could be solutions for that. We must be creative, absolutely. Please. So I have 1, 2, 3, 4, 6 hands. I don't know which was first. I see you. Yeah, please.
Thank you. I just wanted to go back to what Ralph was starting to say, which is really a practical way to identify some topic, for the next meeting. And something I'd like to ask you is, in addition to the person that are seated in here, you mentioned that we should be considering what accelerating is doing, how we can contribute in holding the right stakeholder in the same place. So what EMA can do, but also if you can tell us what we can do in order to convene the appropriate stakeholders, and happy to start with the kind of topic that you suggested. So I'm really trying to move a little bit further to see who else, how we can contribute in bringing, the right person, and happy to set, a specific agenda, maybe then to see what additional documents or research we need to do to really have the discussion.
You're right, but we will follow up with a survey you will receive, and where we'll have the opportunity of any suggestion we believe is valid. So I have 1, 2, 4, 1, 2, 3, and so Josh, please.
Yes, thank you. I think more than anything, I just want to thank and commend everyone here, but in particular the EMA. It's really evident that over the last decade, at least it's my perception, that there has been a real evolution in thinking, and just a forward thinking and a willingness to be adaptable, to, as Francesco said, to be mud diggers. And I think to deviate from tradition and dogma in a way that I did not hear a decade ago when we last had interactions with the EMA. I think that your thinking at this point is far ahead of where industry and the clinician community understand your thinking to be. And I think that unfortunately, there's a perception of a lack of flexibility, a lack of adaptability, a lack of forward thinking, and that has a chilling effect, unfortunately, on trial design and on investment. And so if there was some way for what has been expressed in this room and what hopefully will result from the conversations that will ensue to be made public and disseminated in some way, I think that that will have a tremendous impact on unlocking investment and unlocking initiative on the part of clinicians and the patient community as well. And so my question is there some way that plans could be made for the results of these conversations to be made public, to be disseminated in some fashion?
Sure, and Karin will explain, or Sylvia.
I mean, I don't know if I explained well, but for sure the idea is to keep notes of everything was said. We have also recorded the meeting, so it's not going to be difficult, and to come out with a paper that can summarize the most important aspect that were touched today. And then for sure we have the option for a follow-up meeting, right? So the idea is that we might try even all together to identify some more interesting general topic that can affect sarcoma and maybe not only sarcoma, and come back for another workshop like this one before summer.
Yeah, that's
correct.
That's wonderful. Just to be specific, we've kind of punted specific questions to follow-up conversations. And I would just hope that those follow-up conversations could have some level of openness and some plan for dissemination of specific answers, not just kind of general questions. So I appreciate very much the spirit of openness and dissemination.
Thanks a lot. So others? Yes, please.
So thank you. First of all, I would like to compliment this great meeting and really thank you for so many great discussions. At the beginning, we were also talking about compassionate use and also about a little bit more from the regulatory perspective. And I approached Mrs. Valerie Dennis. We had a very great discussions about what is going on in France in terms of certain regulations. But in fact, I'm pretty curious, how about other member states? How about Germany, or what's going on in here in the Netherlands, etc., etc. So maybe another suggestion for other topics which could be relevant within this setting. Thank you.
Yeah, for sure. I fully agree with you. This should be the topic of another meeting, for sure. Piotr, we can never say a surgeon not to talk.
Okay, thank you. Very shortly. So because the Europe's cancer beating plan, of course, includes digitalization. So I think that it should be also the pressure on the state countries that implementation of the registries in the national routine health systems should be done, especially in oncology. So it really can resolve a lot of problems, because we are opening different registries and we are lacking funding, lacking interest, lacking the long-term follow-up. This is the major issue. Now it could be done really in much more automatical way. And I think that it should be the discussion from EMA that it should be something which is obligatory now when we are implementing the Europe's cancer beating plan. And I think that it can resolve a lot of issue. Maybe it will be not 100% reliable, but in the mass of them information, we can get a very, very important conclusion as a natural history of the disease, etc. So.
No, thanks for agreement. For your information, there are initiatives at the level of the European Commission to support a more efficient use of individual data that could be collected throughout Europe. But this could be a topic for next meeting. If you allow me, I will like to jump to the conclusions that I elaborated for this meeting. Not conclusions, but projects more than conclusions. The take-home message should be, we need standardized, high-quality, collections of clinical biological data, and this is up to the investigators to do so with our help to be sure that this meets the regulatory requirements. But this is something we will have to discuss on a case-by-case basis, and we must, it is our duty, offer a forum for such discussions on a case-by-case basis. Which are the endpoints we need? How we could rely on external data? How could we collect prospective data? Is a given registry the best one? Is a given prospective collection organized the way we would like to see it, corresponding to some given projects? So this would be the first bullet point. The second bullet point is for the regulators. We will engage discussions to find the most flexible and adaptive solution to organize these meetings. It cannot be the classical scientific advices. It is not the prime process. It is not the ITF. We have some tools, but we must combine these tools to create something which must not be as rigid as the other tools, especially. So please trust us. We are reflecting actively on that, and the reflection starting on Monday next week, if not on this evening, in my head. The third point would be solutions should be found for those more than orphan conditions, because we say that orphan conditions are conditions that are too rare to have industry interested in developing, but in the end we find industry ready to develop. So this is more than orphan, because sometimes industry is not even interested. So we must find solutions. Article 48 could be a solution, but we must be creative to avoid the very disappointing results of a good question that would benefit the patients in a crucial condition with a very poor prognosis. And the right questions have been addressed by the right development. And in the end, nobody will take in charge the treatment to put it in the market. And the solution is not available. This is a pity. We must find a solution, absolutely. This is a bit more long-term project because the laws do not change that easily. But this is something we have to solve, absolutely, in many situations described today. So we will have how to create a framework for developing rare cancer medicines. We will consult. We won't come with a solution which will not meet your requirements. So we will, if whichever the solution we find, it will be submitted to your suggestions and your approval. And the solution for that is a survey that will be sent by next week, where you will have an opportunity to express what your priorities are, what your suggestions could be, and it will be the first step to engage into a dialogue to produce more efficient ambient to better solutions to our problems, our meaning problems of academics, of patients, and of regulators, but of patients in the first place. So it would be my conclusion. If you have any burning comments before I leave, because I had to leave at 5 sharp, but I extended already. Sorry for that.
Maybe I just add 2 words from the other side, even though we are all sitting around. So meaning that we are all on the same side in a way. So I again want to express the gratitude of all of us for starting initiatives like the repurposing. That was basically the one who brought us here as EORTC and as the sarcoma community. we are very happy to listen that we can work together. And as Josh was saying, sometimes maybe the perception is that this is not that easy. So it's good to listen that new way to get in contact, a new opportunity for meeting, different, kind of meeting will be conceived. As a sarcoma community, I think our, intention is to move forward developing our global project, our global collaboration. PUSH is one of these projects, but then we are working also at several different levels. And I'm happy to say that we can consider you for the future projects and what is started and is starting as an observer and advisor that we can, bring in to try to optimize what we do and to take the best from all the energy and the enthusiasm and the work that we can do together. So with that, also thanks to all those who are connected virtually and those who will, as we said, try to make the best use of what we said today. We will work all together on the summary and the report of what we have done. So thank you.
Thank you.